The chainsaw cuts through the frozen earth of Siberia like a surgeon's scalpel, revealing what lies beneath 40,000 years of permafrost. Dr. Evgeny Mashenko kneels beside the excavation site, his breath forming clouds in the minus 30-degree air as he carefully brushes away ice crystals from what appears to be a section of reddish-brown fur. This is not just another archaeological dig. This is a crime scene that has been perfectly preserved since the day the victim died, and the evidence it contains is about to rewrite everything we thought we knew about the greatest extinction mystery in human history. The creature emerging from the ice is a woolly mammoth, but this is no museum skeleton or artistic reconstruction. This is Lyuba, a baby mammoth so perfectly preserved that her last meal is still visible in her stomach, her trunk still curled in the position she held when she took her final breath over 40,000 years ago. Her dark eyes, though clouded by millennia, seem to stare accusingly at the scientists who have disturbed her eternal rest. She is not just a fossil. She is evidence in a case that has baffled investigators for over a century. Lyuba represents one victim in what may be the largest mass extinction event in recent geological history. 10,000 years ago, millions of woolly mammoths roamed across the northern hemisphere, from the frozen tundra of Siberia to the grasslands of Alaska, from the plains of Europe to the steppes of Central Asia. They were giants that had dominated their world for over 400,000 years, surviving multiple ice ages, volcanic eruptions, and dramatic climate shifts that had eliminated countless other species. Then, in what amounts to an evolutionary blink of an eye, they were gone. The crime scene extends across an entire hemisphere. In the permanently frozen soils of Siberia, scientists have discovered mammoth carcasses by the thousands, their bodies scattered across the landscape like victims of some ancient catastrophe. In Alaska, mammoth bones emerge from riverbanks with such regularity that local residents use them as doorstops and garden decorations. Across Europe and Asia, excavations reveal kill sites where mammoth bones are mixed with human artifacts, spear points, and the charred remains of ancient campfires. The investigation into mammoth extinction has all the elements of a classic murder mystery. There are multiple suspects, each with clear motive and opportunity. There is conflicting evidence that seems to point in different directions depending on how it is interpreted. There are expert witnesses whose testimony contradicts each other, and there is a timeline that refuses to make sense no matter how many times investigators try to piece it together. The first suspect is climate change. Approximately 12,000 years ago, the Earth was emerging from the last ice age in a period of rapid warming that transformed the entire northern hemisphere. The Mammoth Steppe, a vast grassland ecosystem that had covered millions of square kilometers from western Europe to eastern Alaska, began to disappear as rising temperatures turned frozen tundra into forests and wetlands. For creatures that had evolved to thrive in cold, dry conditions, this environmental transformation was nothing short of apocalyptic. The second suspect is humanity. Modern humans had been spreading across the globe for tens of thousands of years, armed with increasingly sophisticated hunting technologies and an appetite for megafauna that seemed to leave extinction in their wake. Wherever humans went, large animals disappeared. Australia lost its giant marsupials. The Americas lost their ground sloths and giant bears. Europe lost its cave lions and woolly rhinoceros. The pattern was so consistent that it seemed to point to a single cause: us. The third suspect is disease. As humans spread across the globe, they carried with them pathogens from Africa that had never before encountered the immune systems of animals in other continents. These diseases could have spread through mammoth populations like wildfire, killing millions without leaving any direct evidence in the fossil record. It was the perfect crime, one that would leave investigators searching for clues that had long since decomposed. Each suspect has alibis that complicate the case. Climate change had occurred many times before without driving mammoths to extinction. Humans had been living alongside mammoths for hundreds of thousands of years without eliminating them. Diseases capable of causing such widespread mortality would likely have affected other species as well. Yet many animals survived the same period that saw mammoth extinction. The evidence preservation in this case is unprecedented in forensic science. The permafrost of Siberia has created conditions that rival the best crime labs in the world, maintaining DNA, proteins, and even cellular structures for tens of thousands of years. Scientists can examine not just the bones of extinct mammoths, but their skin, their hair, their internal organs, and even the contents of their stomachs. They can extract genetic material that reveals population sizes, migration patterns, and family relationships. They can analyze isotopes in mammoth teeth that show what these animals were eating in the months before they died. Modern investigative techniques have revealed details about mammoth biology and behavior that would have been impossible to discover from fossil bones alone. We know that mammoths had complex social structures led by experienced matriarchs who remembered the locations of water sources and migration routes across vast territories. We know that they used their tusks not just for defense, but as tools for clearing snow and stripping bark from trees. We know that they could communicate across long distances using infrasonic calls too low for human ears to detect. The genetic evidence has revealed that mammoths were not a single species, but a complex group of related populations that had been diverging for hundreds of thousands of years. Some populations were adapting to life in forests, others to island environments, and still others to the changing conditions at the edge of their range. This diversity should have provided insurance against extinction, yet somehow all of these different mammoth lineages disappeared within the same narrow window of time. The timeline of mammoth extinction varies by location in ways that may provide crucial clues about the cause. In some regions, mammoths disappeared abruptly, as if some catastrophic event had eliminated entire populations overnight. In others, the decline was gradual, with populations shrinking over thousands of years before finally winking out. The last mammoths survived on isolated islands, where they persisted for thousands of years after their mainland relatives had vanished, suggesting that isolation itself might provide protection against whatever force was driving the extinction. Yet not all large animals disappeared. Musk oxen, caribou, bison, and many other species that shared habitats with mammoths managed to survive the same period that saw mammoth extinction. This selective pattern of survival and extinction provides additional clues but also deepens the mystery. What made mammoths vulnerable when their neighbors proved resilient? The investigation continues today in laboratories around the world, where scientists are using cutting-edge techniques to extract new evidence from ancient remains. Advances in DNA analysis are revealing population histories that span hundreds of thousands of years. Isotope analysis is providing detailed information about mammoth diet and migration patterns. Computer modeling is testing different extinction scenarios to see which ones match the pattern of evidence. The stakes in solving this case extend far beyond academic curiosity. The same forces that drove mammoth extinction may be operating today, threatening modern elephant populations and countless other species around the world. Understanding what happened to the mammoths could provide crucial insights for preventing similar extinctions in the future. The Pleistocene crime scene holds lessons that could determine whether elephants, rhinos, and other megafauna survive the current century. The autopsy table is not made of steel, but of ancient permafrost, and the body lying upon it has been waiting for this examination for over 40,000 years. Dr. Daniel Fisher runs his gloved hands over the perfectly preserved remains of a male woolly mammoth discovered in the Yamal Peninsula, his fingers tracing the contours of muscles that still retain their original texture beneath layers of reddish-brown fur. This is not just any victim. This is the most complete mammoth specimen ever recovered, and his story is about to reveal the shocking truth about what life was really like for these giants before their mysterious disappearance from the earth. The victim's name, assigned by the scientific team, is Genya, and his biography reads like that of a successful patriarch who had everything to live for when death came calling. At the time of his demise, Genya was approximately 45 years old, in the prime of his life for a species that could live well into their 60s. He stood 4.5 meters tall at the shoulder, weighed approximately 6 tons, and possessed tusks that curved gracefully outward for nearly 4 meters, indicating a lifetime of successful foraging and social dominance that had allowed him to grow these magnificent ivory weapons to their full potential. The forensic examination reveals a creature so perfectly adapted to his environment that extinction should have been impossible. Genya's body tells the story of evolutionary engineering that took millions of years to perfect. His skin, preserved in the permafrost like leather treated by the world's most skilled craftsmen, shows the dual-layer fur system that made woolly mammoths masters of the Ice Age world. The outer layer consists of guard hairs up to 90 centimeters long, creating a waterproof barrier that could shed rain and snow while trapping warm air beneath. Underneath, a dense undercoat of fine wool provided insulation so effective that mammoths could maintain core body temperatures even when outside temperatures dropped to minus 40 degrees Celsius. The victim's skull reveals a brain case large enough to house an organ that weighed over 5 kilograms, indicating intellectual capabilities that rivaled those of modern elephants. Recent studies of mammoth behavior, reconstructed from fossil evidence and DNA analysis, suggest these were highly intelligent animals capable of complex problem-solving, long-term memory, and sophisticated social interactions. They could remember the locations of water sources and seasonal feeding grounds across territories that spanned hundreds of kilometers, passing this knowledge down through generations in cultural traditions that persisted for thousands of years. Genya's tusks tell their own story of a life lived successfully in one of Earth's most challenging environments. Growth rings in the ivory, like tree rings, reveal periods of plenty and scarcity throughout his 45 years of life. The early rings show rapid growth during his youth, when mammoth populations were stable and food was abundant. The middle rings document his prime years, when he would have competed with other males for mating rights and established his position in the complex social hierarchy that governed mammoth society. The outer rings formed in his final years show no signs of the stress that would indicate environmental collapse or resource scarcity. The stomach contents preserved within Genya's digestive system provide a window into the mammoth world that existed just before the extinction event. His last meal consisted of grasses, sedges, and herbs that grew in the mammoth steppe, the vast grassland ecosystem that supported the largest herds of large mammals that Earth has ever known. Analysis of pollen grains and plant fragments found in his stomach reveals a diverse diet that included over 30 different plant species, indicating that his environment was rich and varied rather than the barren wasteland that some theories suggest existed at the time of mammoth extinction. The mammoth steppe itself was one of the most productive ecosystems in Earth's history, a cold-adapted grassland that stretched from Western Europe across Siberia to Alaska and down into the northern regions of North America. This was not the frozen tundra that exists in these regions today, but a rich grassland maintained by the grazing activities of millions of large herbivores, including mammoths, woolly rhinoceros, steppe bison, and wild horses. The grasses that grew in this ecosystem were specially adapted to cold, dry conditions, remaining nutritious even when frozen and providing high-energy food that could sustain massive animals through long Arctic winters. Genya would have lived in a matriarchal society similar to that of modern elephants, where family groups consisting of related females and their offspring were led by the oldest and most experienced female. Adult males like Genya typically lived more solitary lives, joining family groups only during mating season or when environmental conditions made cooperation necessary for survival. The social bonds between mammoth family members were strong and long-lasting, with evidence suggesting that mammoths grieved for their dead and showed concern for injured or distressed group members. The victim's feet reveal another marvel of evolutionary adaptation that made mammoths such successful inhabitants of frozen landscapes. Each foot was equipped with a unique cushioning system consisting of fatty tissue that acted like a natural snowshoe, distributing the animal's enormous weight across a larger surface area and preventing them from sinking into snow or soft ground. The soles of their feet contained specialized tissue that could detect vibrations through the ground, allowing mammoths to communicate with distant family groups through seismic signals and to sense the approach of predators or the location of water sources buried beneath snow. The forensic analysis of Genya's teeth provides crucial evidence about mammoth feeding behavior and the environmental conditions they faced. Unlike modern elephants, which replace their teeth continuously throughout their lives, mammoths had only 6 sets of teeth that had to last their entire lifetime. The wear patterns on Genya's teeth show that he had progressed to his 5th set, indicating that he was an older individual, but still had years of life ahead of him when death occurred. The grinding surfaces of these teeth were perfectly adapted for processing the tough, fibrous grasses of the mammoth steppe, with complex ridges and valleys that could break down plant material into digestible fragments. Chemical analysis of the victim's bones reveals information about his migration patterns and seasonal movements throughout his lifetime. Isotope ratios in the bone tissue change depending on the types of food consumed and the water sources used, creating a chemical signature that can be traced back to specific geographic regions. Genia's bones indicate that he had traveled extensively throughout his life, covering distances of hundreds of kilometers as he followed seasonal patterns of plant growth and sought out the best feeding areas available in his vast territory. The reproductive history encoded in Genia's remains tells the story of a successful breeding male who had likely fathered numerous offspring during his prime years. Genetic analysis of mammoth populations suggests that successful males like Genia could father dozens of calves over their lifetime, contributing significantly to the genetic diversity and stability of their populations. The slow reproductive rate of mammoths, with females giving birth to a single calf only every 3 to 6 years after a 22-month gestation period, meant that every successful breeding male played a crucial role in maintaining population numbers. The victim's ear structure reveals adaptations that allowed mammoths to communicate across the vast distances of the mammoth steppe, using infrasonic calls similar to those used by modern elephants. These low-frequency vocalizations could travel for dozens of kilometers, allowing family groups to coordinate their movements and maintain contact even when separated by great distances. The ability to communicate over long ranges was essential for animals that needed to exploit scattered resources across enormous territories and coordinate their movements with changing seasonal conditions. Evidence from Genia's nasal cavity shows adaptations that allowed mammoths to survive in environments where the air temperature could reach life-threatening levels of cold. The mammoth's trunk contained a complex system of blood vessels and air passages that pre-warmed incoming air before it reached the lungs, preventing damage to sensitive respiratory tissues. The trunk also functioned as a multi-purpose tool that could be used for gathering food, manipulating objects, communicating through touch, and even as a snorkel when crossing rivers or deep snow. The social dynamics of mammoth herds can be reconstructed from the distribution of mammoth remains across the landscape and the age and sex patterns found at different sites. Family groups typically consisted of 8 to 12 individuals, including adult females, their daughters, and calves of various ages. These groups maintained territories that overlapped with those of related families, creating larger social networks that could include dozens of individuals who recognized and cooperated with each other across multiple generations. The seasonal migrations that structured mammoth society required extensive environmental knowledge and sophisticated navigation abilities. Herds followed ancient pathways that had been established over thousands of years, timing their movements to take advantage of peak growing seasons in different regions and to avoid areas where conditions were unsuitable for feeding or raising young. The leadership of experienced matriarchs was crucial for these migrations, as they possessed the accumulated knowledge of decades of successful journeys and could guide their families to safety even during years when environmental conditions were challenging. The victim's immune system, reconstructed through analysis of genetic material found in his remains, reveals a species that had successfully adapted to the disease challenges of their environment over hundreds of thousands of years. Mammoth populations had developed resistance to parasites and pathogens specific to cold environments, and their genetic diversity suggested that they maintained robust immune responses that could cope with new disease challenges as they arose. The evidence from Genia's remains paints a picture of a species at the height of evolutionary success when extinction occurred. These were not struggling animals barely clinging to survival in a deteriorating environment, but thriving giants who dominated their ecosystem and showed every indication of continued success. The Mammoth steppe supported some of the highest biomass densities of large mammals ever recorded on Earth, with recent estimates suggesting that the Northern Hemisphere may have supported over 5 million woolly mammoths at the peak of their abundance. The mystery deepens when we consider that mammoths had successfully survived previous episodes of climate change that were equally dramatic as the warming that occurred at the end of the last ice age. During their 400,000-year history, mammoth populations had weathered multiple glacial cycles, volcanic eruptions, and environmental disruptions that eliminated many other species. They had proven their adaptability by expanding their range across multiple continents and diversifying into different ecological niches as opportunities arose. The lifestyle revealed by forensic analysis of mammoth remains shows animals that were not only physically robust, but also behaviorally flexible and socially sophisticated. They could modify their behavior in response to changing conditions, exploit new food sources when traditional ones became unavailable, and maintain social cohesion even under stressful circumstances. These characteristics should have provided insurance against extinction, yet somehow proved insufficient when the final crisis arrived. The examination of Genia's remains raises disturbing questions about what force could have been powerful enough to eliminate such successful and well-adapted animals. The evidence suggests that mammoth extinction was not the result of gradual decline due to environmental stress, but rather a rapid collapse that occurred despite the species' apparent health and stability. This pattern points toward an external cause that was both sudden and overwhelming, something that struck before mammoths had time to adapt their successful survival strategies to meet a new and unprecedented threat. The forensic evidence from this remarkable victim demands that we look beyond simple explanations for mammoth extinction and consider more complex scenarios involving multiple interacting factors. Genia's story is not one of a species in decline, but of a successful giant struck down in his prime by forces that remain to be identified. The investigation must now turn to examining the suspects who might have had both the motive and the means to commit such a massive crime against one of evolution's greatest success stories. The interrogation room is a sterile laboratory where ice cores from Greenland stand like silent witnesses in refrigerated storage units, each one containing evidence that could either convict or exonerate our first suspect in the mammoth extinction case. Dr. Anders Svensson adjusts his microscope as he prepares to examine testimony that has been locked in ice for over 12,000 years. Testimony that will reveal whether climate change should be charged with the greatest extinction crime in recent history. Suspect number one sits in the dock not as a person, but as a force of nature so powerful that it has the ability to reshape entire continents. Climate change, specifically the dramatic warming that marked the end of the last ice age, has motive, opportunity, and a track record of environmental destruction that spans geological time. The evidence against this suspect is overwhelming in its scope and undeniable in its timing. Yet the defense maintains that previous interactions between climate and mammoths had always resulted in adaptation rather than extinction. The motive is crystal clear: habitat destruction on a scale that defies human comprehension. 12,000 years ago, the Earth began emerging from the last ice age in a period of warming so rapid and dramatic that it transformed the entire northern hemisphere within a few millennia. Temperatures rose by 5 to 10 degrees Celsius. Ice sheets that had covered millions of square kilometers for tens of thousands of years began melting at unprecedented rates, and sea levels rose by over 100 meters as glacial meltwater poured into the oceans. For mammoths, this environmental transformation was nothing short of apocalyptic. The mammoth steppe, the vast grassland ecosystem that had been their home for hundreds of thousands of years, began disappearing as warmer temperatures and increased precipitation created conditions that favored forests and wetlands over the cold, dry grasslands that mammoths needed to survive. In regions where mammoth herds had grazed for countless generations, trees began growing for the first time in millennia, fundamentally altering the character of landscapes that had remained stable throughout mammoth evolutionary history. The opportunity was perfect in its timing and global in its reach. Unlike previous climate changes that had occurred gradually over tens of thousands of years, the warming at the end of the last ice age happened with shocking rapidity. Ice core data from Greenland reveals that in some regions, temperatures rose by several degrees within decades, creating environmental changes that occurred faster than mammoth generations could adapt. This was not the gradual warming that had characterized previous interglacial periods, but a climate shift so rapid that it resembled a natural catastrophe rather than normal environmental variation. The geographic scope of the climate change provides the suspect with an unshakable alibi for being present at every crime scene simultaneously. From Siberia to Alaska, from northern Europe to the Canadian Arctic, the same pattern of rapid warming and environmental transformation was occurring across the entire mammoth range during the same narrow window of time when mammoth populations collapsed. No other suspect in the case can claim such comprehensive presence at all locations where mammoth extinction occurred. The method of killing reveals climate change as a suspect capable of murder through habitat transformation rather than direct violence. As temperatures rose and precipitation patterns changed, the plant communities that form the foundation of mammoth ecosystems began shifting from cold-adapted grasses to warm-adapted trees and shrubs. The grasses that mammoths had evolved to eat over millions of years were replaced by vegetation that was difficult or impossible for mammoths to digest, creating a form of starvation that occurred not because food was absent, but because the available food was unsuitable. The forensic evidence preserved in ice cores provides a detailed timeline of the climate changes that occurred during the period of mammoth extinction. These ice cores, drilled from the Greenland ice sheet and other glacial sites around the Arctic, contain annual layers that preserve atmospheric conditions with the precision of a geological calendar. Analysis of gases trapped in ice bubbles reveals carbon dioxide levels, volcanic ash layers document major eruptions, and isotope ratios provide information about temperature and precipitation patterns with year-by-year resolution. The testimony from these ice core witnesses reveals that the warming began approximately 15,000 years ago and accelerated dramatically around 12,000 years ago, precisely matching the timeline of mammoth population decline documented in the fossil record. The correlation is so precise that it provides compelling circumstantial evidence for climate change as the primary cause of mammoth extinction. The probability that such perfect timing could be coincidental is so low that it suggests a direct causal relationship between environmental change and species collapse. However, the defense presents equally compelling evidence that challenges the prosecution's case against climate change. The most damaging piece of evidence for the prosecution is the fact that mammoths had successfully survived multiple previous episodes of climate change that were equally dramatic as the warming that occurred at the end of the last ice age. During their 400,000-year evolutionary history, mammoth populations had weathered at least 4 major glacial cycles, each involving temperature changes of similar magnitude to those that occurred during the suspected extinction event. Furthermore, the prosecution counters with evidence that the climate change at the end of the last ice age was fundamentally different from previous warming episodes in ways that made mammoth adaptation impossible. Unlike earlier climate transitions that had occurred gradually over tens of thousands of years, the final warming was compressed into a much shorter time period that did not allow sufficient time for evolutionary adaptation. Additionally, the magnitude of environmental change was greater than previous transitions, with some regions experiencing temperature increases that exceeded anything in the previous mammoth evolutionary record. The prosecutor presents detailed analysis of pollen records from across the Mammoth Range, showing that the vegetation changes that occurred during the final warming were more rapid and extensive than those documented for previous climate transitions. In some regions, the transition from mammoth steppe to forest occurred within a few centuries, creating environmental changes that happened faster than mammoth reproductive cycles could respond through natural selection. The ice core evidence also reveals that the final warming was accompanied by increased climatic instability that would have made adaptation particularly difficult. Rather than a smooth transition from cold to warm conditions, the climate record shows rapid fluctuations that would have prevented mammoth populations from establishing stable adaptations to new environmental conditions. This climatic instability, documented in ice core records as repeated temperature swings of several degrees within decades, created a moving target that made successful adaptation virtually impossible. The prosecution introduces evidence from other species that survived the same climate changes that allegedly killed the mammoths, arguing that selective extinction patterns support the case against climate change as the sole cause of mammoth disappearance. Caribou, musk oxen, and Arctic foxes all survived the same environmental transitions that supposedly eliminated mammoths, suggesting that climate change alone cannot explain the extinction pattern. If environmental change was the primary cause, then all large Arctic mammals should have disappeared rather than just selected species. However, the defense argues that mammoths were uniquely vulnerable to the specific type of environmental change that occurred at the end of the Ice Age. As the largest herbivores in their ecosystem, mammoths had enormous daily food requirements that made them particularly sensitive to changes in vegetation productivity and composition. While smaller animals could survive on marginal food sources or switch to alternative diets, Mammoths needed massive quantities of high-quality vegetation that simply was not available in the transformed post-glacial landscapes. The prosecution presents compelling evidence from mammoth tooth analysis that shows dietary stress in populations living during the transition period. Isotope analysis of mammoth teeth reveals that animals living during the final millennia of mammoth existence were consuming lower quality food than their ancestors, indicating that environmental degradation was already affecting mammoth health before extinction occurred. This evidence suggests that climate change was slowly weakening mammoth populations even before other factors delivered the final blow. Chemical analysis of mammoth bones from the extinction period reveals elevated stress hormones that indicate populations were experiencing chronic physiological stress during their final years. This stress response is consistent with animals struggling to cope with environmental conditions that exceeded their adaptive capacity, supporting the prosecution's argument that climate change was placing unsustainable pressure on mammoth populations. The defense introduces evidence from mammoth DNA that reveals increased genetic diversity in some populations during the period leading up to extinction, suggesting that gene flow between previously isolated populations was increasing rather than decreasing during the climate transition. This evidence contradicts the prosecution's argument that environmental change was fragmenting mammoth populations and reducing their adaptive potential. The prosecution responds with evidence from modern climate science showing that the rate of environmental change at the end of the last ice age was unprecedented in mammoth evolutionary history. Computer models of ice age climate demonstrate that previous transitions had occurred gradually over periods of 10 to 20,000 years, while the final warming occurred within 2 to 3,000 years. This acceleration of environmental change compressed adaptive pressures into time periods that were too short for evolutionary responses to develop. The ice core testimony reveals additional evidence that supports the prosecution's case, including documentation of rapid changes in atmospheric circulation patterns that would have disrupted the seasonal cycles that mammoth migrations depended upon. Changes in wind patterns and storm tracks altered precipitation distribution across the mammoth range, creating unpredictable resource availability that would have made traditional mammoth survival strategies ineffective. The prosecution presents evidence that the climate changes at the end of the ice age were accompanied by increased frequency of extreme weather events that would have posed additional challenges for mammoth populations. Ice core records document increased volcanic activity during this period, which would have caused temporary cooling episodes that disrupted plant growth and created additional environmental stress for animals already struggling with long-term warming trends. However, the defense argues that the correlation between climate change and mammoth extinction, while compelling, does not constitute proof of causation. The defense points to regions where mammoth populations persisted for thousands of years after the main extinction event, including island populations that survived until as recently as 4,000 years ago. These surviving populations experienced the same climate changes as their extinct relatives, yet managed to persist in stable numbers for millennia after the supposed climate-driven extinction. The defense introduces expert testimony from population biologists who argue that climate change alone would have caused gradual population decline rather than the rapid collapse that characterizes mammoth extinction. Mathematical models of population dynamics under environmental stress predict that climate change should have caused mammoth numbers to decline over tens of thousands of years rather than disappearing within a few millennia. The prosecution counters with evidence that climate change created a cascade of environmental effects that went beyond simple temperature increases to include fundamental reorganization of Arctic ecosystems. The replacement of mammoth steppe with forests and wetlands created environments that were not just unsuitable for mammoths, but actively hostile to their survival. Forests restricted movement patterns that had been essential for mammoth foraging strategies, while wetlands created obstacles that could trap and kill animals that had evolved for life on solid ground. The ice core evidence reveals that the warming at the end of the ice age was accompanied by dramatic changes in seasonal patterns that would have disrupted mammoth reproductive cycles and migration timing. Changes in the length and intensity of winter seasons altered the timing of plant growth cycles, creating mismatches between mammoth calving seasons and peak food availability that would have reduced reproductive success and increased calf mortality. The prosecution presents final evidence from oxygen isotope analysis of mammoth teeth that reveals physiological stress indicators in the final mammoth populations, suggesting that animals were struggling with thermal regulation in the warmer post-glacial environment. The thick fur and large body size that had made mammoths masters of the Ice Age became liabilities in the warming world, creating heat stress that reduced fitness and reproductive success, even in populations that had access to adequate food resources. As the interrogation of climate change concludes, the evidence presents a complex picture that supports both prosecution and defense arguments. The timing and geographic scope of climate change provide compelling, circumstantial evidence for its role in mammoth extinction, yet the survival of mammoths during previous climate transitions and the persistence of some populations long after the main extinction event create reasonable doubt about whether environmental change alone was sufficient to cause the disappearance of such successful and adaptable animals. The case against climate change remains strong but not conclusive, leaving room for additional suspects to be examined before a final verdict can be reached. The evidence locker contains artifacts that would make any prosecutor's case: stone spear points still embedded in mammoth ribs, butchering marks carved into ancient bones, and archaeological sites where mammoth remains are mixed with human tools in patterns that can only mean one thing. Dr. Gary Haynes closes the file folder containing 40 years of research as he prepares to present the case against humanity's most controversial suspect in the mammoth extinction trial. This is not just another scientific debate. This is an indictment of our own species for committing what may be the first recorded genocide in human history. Suspect number 2 enters the interrogation room not as an individual, but as an entire species whose expansion across the globe coincides so perfectly with megafauna extinctions that the correlation appears to be more than coincidence. Homo sapiens, armed with increasingly sophisticated hunting technologies and an appetite for large prey that seems to leave extinction in their wake, stands accused of systematically eliminating mammoth populations across 2 continents through a campaign of overexploitation that lasted thousands of years. The motive is as old as humanity itself: survival through resource acquisition. For early human populations struggling to establish themselves in the challenging environments of Ice Age Europe and North America, mammoths represented the ultimate prize in a landscape where calories could mean the difference between survival and death. A single adult mammoth could provide enough meat to feed a band of 100 people for an entire month, along with hides for clothing and shelter, bones for tools and fuel, and ivory for weapons and trade goods that could be exchanged across vast distances. The psychological profile of the suspect reveals a species unlike any predator that had previously existed on Earth. Humans possessed intelligence that allowed them to develop complex hunting strategies, communication abilities that enabled coordinated group attacks, and technological innovations that gave them weapons far more effective than claws or teeth. Most importantly, humans had the cultural capacity to accumulate and transmit hunting knowledge across generations, creating an ever-improving arsenal of techniques for killing animals that had no evolutionary experience with such a formidable adversary. The opportunity begins to emerge in the archaeological record approximately 45,000 years ago, when the first modern humans appeared in Europe carrying advanced stone tools and sophisticated hunting equipment. Unlike previous human species that had coexisted with mammoths for hundreds of thousands of years without causing extinctions, these new arrivals brought revolutionary technologies and hunting strategies that transformed them from minor predators into the most dangerous threat that mammoth populations had ever faced. The timeline of human expansion across the Northern Hemisphere reads like a case study in serial extinction. In Europe, mammoth populations began declining shortly after the arrival of modern humans, despite environmental conditions that remained favorable for mammoth survival. In North America, the appearance of the Clovis culture 13,000 years ago coincides almost exactly with the final collapse of mammoth populations that had thrived on the continent for hundreds of thousands of years. The weapon technology possessed by these human populations represents a quantum leap in hunting effectiveness that gave them unprecedented power over their prey. The atlatl, a spear-throwing device that could hurl projectiles at speeds exceeding 100 kilometers per hour, transformed humans from close-range hunters who risked injury with every kill into long-distance predators who could bring down massive animals while remaining safely beyond the reach of tusks and trampling feet. The Clovis point technology that appeared in North America represents perhaps the most sophisticated hunting tool ever developed for killing megafauna. These finely crafted stone projectiles were designed specifically for penetrating the thick hides of large animals, with fluted bases that allowed deep penetration and razor-sharp edges that could cause massive internal bleeding. Archaeological experiments have demonstrated that Clovis points could penetrate mammoth hide and deliver fatal wounds with a single well-placed strike. The prosecution presents evidence from dozens of kill sites across North America where mammoth bones are found in direct association with Clovis points and other human artifacts. At Blackwater Draw in New Mexico, Clovis points were discovered embedded in mammoth vertebrae in positions that could only have resulted from human hunting activities. At Murray Springs in Arizona, mammoth bones show clear evidence of butchering with stone tools, while the arrangement of bones suggests systematic processing for meat, hide, and other valuable materials. The forensic analysis of these kill sites reveals hunting strategies that demonstrate remarkable sophistication and planning. Mammoth bones are often found at the bottoms of arroyos and other natural traps, suggesting that human hunters use their knowledge of terrain and mammoth behavior to drive animals into locations where escape would be impossible. The distribution of bones and artifacts at these sites indicates that hunting was a coordinated group activity that required precise timing and communication between multiple participants. The overkill hypothesis, first proposed by paleontologist Paul Martin in 1967, argues that human hunting was not just a contributing factor in mammoth extinction, but the primary cause. Martin's mathematical models demonstrate that even modest levels of human hunting pressure could drive mammoth populations to extinction within a few centuries, particularly if hunters preferentially targeted female and young animals that were essential for population reproduction. The behavioral analysis of human hunting patterns supports the overkill hypothesis by revealing hunting strategies that were unsustainable over the long term. Archaeological evidence suggests that human hunters often killed more animals than they could use, leaving partially processed carcasses scattered across the landscape in patterns that indicate wasteful rather than conservative resource utilization. This pattern of overexploitation is consistent with the behavior of a newly arrived predator that had not yet learned the sustainable hunting practices necessary for long-term coexistence with prey species. The prosecution introduces evidence from other continents where human arrival coincides with megafauna extinctions in patterns that are virtually identical to what occurred in North America. In Australia, the arrival of humans approximately 50,000 years ago was followed by the extinction of giant marsupials, flightless birds, and other megafauna that had thrived for millions of years. In Madagascar, human colonization 2,000 years ago resulted in the rapid extinction of elephant birds, giant lemurs, and hippopotamuses that had survived until human contact. The global pattern of megafauna extinctions following human expansion provides compelling circumstantial evidence for human culpability in mammoth extinction. The consistency of this pattern across multiple continents and different time periods suggests that human arrival consistently triggers ecological disruption that leads to the extinction of large animals that had previously coexisted successfully with other predators and environmental challenges. However, the defense presents equally compelling evidence that challenges the prosecution's case against humanity. The most significant piece of evidence for the defense is the fact that humans and mammoths had coexisted successfully for hundreds of thousands of years without causing extinctions. Neanderthals and other early human species had been hunting mammoths in Europe and Asia for over 200,000 years. Yet mammoth populations remained stable and even expanded their range during periods of human presence. The defense argues that if human hunting alone was sufficient to cause mammoth extinction, then mammoths should have disappeared much earlier when human populations first developed the ability to hunt large animals. The fact that mammoth extinction did not occur until the end of the last ice age, despite centuries of human hunting pressure, suggests that other factors must have been responsible for making mammoth populations suddenly vulnerable to human predation. Furthermore, the defense points to evidence that human hunting of mammoths was often sustainable and carefully managed rather than the reckless overexploitation suggested by the prosecution. Archaeological sites from Siberia and other regions show evidence of mammoth hunting that spans thousands of years without apparent population decline, indicating that human communities had developed cultural practices that prevented overexploitation of their most important resource. The defense introduces expert testimony from anthropologists who argue that indigenous hunting cultures typically develop sophisticated conservation practices that prevent the extinction of prey species upon which they depend. These cultural practices include seasonal hunting restrictions, taboos against killing pregnant females, and territorial systems that prevent overexploitation of local resources. The rapid extinction of mammoths is inconsistent with the conservation practices that characterize sustainable hunting cultures. The prosecution counters with evidence that the humans responsible for mammoth extinction were not established indigenous populations, but newly arrived colonizers who had not yet developed sustainable relationships with their new environment. The Clovis people who appeared in North America 13,000 years ago were expanding rapidly across an unfamiliar continent, encountering prey species that had no evolutionary experience with human predation and therefore lacked appropriate fear responses. The Blitzkrieg model proposed by the prosecution suggests that human expansion across previously uninhabited continents created conditions that were fundamentally different from the long-term coexistence between humans and prey species in Africa and Eurasia. In their original African homeland, prey species had co-evolved with human predators over millions of years, developing behaviors and population strategies that enabled sustainable relationships. When humans expanded into new continents, they encountered naive fauna that lacked these co-evolved defenses. The prosecution presents mathematical models showing that even relatively low levels of human hunting pressure could drive mammoth populations to extinction if the hunting was concentrated on reproductive females and their offspring. Mammoth reproductive biology, with long generation times and low reproductive rates, made populations particularly vulnerable to hunting strategies that targeted breeding animals. The removal of even a small percentage of reproductive females could cause population decline that would accelerate over successive generations. The technological evidence strongly supports the prosecution's case by demonstrating that late Pleistocene humans possessed hunting capabilities that far exceeded those of earlier human populations. The development of composite tools, improved projectile technologies, and coordinated hunting strategies created a quantum leap in human killing efficiency that previous mammoth populations had never experienced. This technological revolution coincides precisely with the timing of mammoth extinction across multiple continents. The prosecution introduces evidence from isotope analysis of human bones that shows late Pleistocene populations in North America had diets that were heavily dependent on megafauna, particularly mammoths. The chemical signatures preserved in human skeletal remains indicate that mammoth meat constituted a substantial portion of Clovis diet, demonstrating both the availability of mammoth prey and the success of human hunting strategies in acquiring it. However, the defense argues that correlation between human diet and mammoth extinction does not prove causation, since human dependence on mammoth meat could reflect opportunistic scavenging rather than active hunting. The defense points to evidence that many supposed kill sites could actually be scavenging sites where humans processed mammoth carcasses that had died from natural causes rather than human predation. The prosecution responds with forensic evidence from mammoth bones that shows clear distinctions between scavenging marks and hunting injuries. Cut marks made by stone tools appear in patterns that are consistent with systematic butchering of fresh carcasses, while the location of spear points in mammoth skeletons indicates wounds that would have been immediately fatal. This evidence demonstrates that humans were actively hunting mammoths rather than simply scavenging dead animals. The defense introduces evidence from populations of mammoths that survived on isolated islands for thousands of years after mainland extinctions, arguing that these island refugia prove that factors other than human hunting were responsible for mammoth disappearance. If humans were the primary cause of extinction, then island populations should have been safe from human predation and should have survived indefinitely. The fact that island mammoths eventually went extinct, despite isolation from human hunters, suggests that other factors were ultimately responsible. The prosecution counters that island mammoth populations were protected only temporarily and eventually succumbed to genetic problems resulting from small population sizes, rather than to the same factors that caused mainland extinctions. The prosecution argues that human hunting reduced mainland mammoth populations to critically low levels, making them vulnerable to other factors that would not have been problematic for healthy populations. The defense presents evidence from computer simulations showing that sustainable human hunting of mammoths was possible under the environmental conditions that existed during the late Pleistocene. These models suggest that careful management of hunting pressure could have allowed mammoth populations to persist indefinitely, contradicting the prosecution's argument that any significant level of human hunting would inevitably lead to extinction. However, the prosecution introduces evidence from behavioral studies showing that newly arrived human populations often fail to develop sustainable hunting practices until after they have caused local extinctions. The pattern of overexploitation followed by resource collapse and cultural adaptation is well documented in historical cases of human colonization, suggesting that the same pattern could have occurred during the initial human expansion into mammoth habitats. The prosecution presents its strongest evidence in the form of the continental pattern of megafauna extinctions that follows human expansion with remarkable consistency. In Africa, where humans and large animals co-evolve together, megafauna diversity remains high despite millions of years of human presence. In Australia, the Americas, and Madagascar, human arrival was followed by rapid megafauna extinction within a few thousand years. This pattern cannot be explained by climate change alone, since all continents experienced similar environmental transitions at the end of the Ice Age. The defense makes its final argument by pointing to the survival of other large herbivores that shared habitats with mammoths, but did not go extinct during the same period. Bison, caribou, and musk oxen all survived the same combination of climate change and human hunting that allegedly killed the mammoths, suggesting that species-specific factors, rather than general hunting pressure or environmental change, were responsible for mammoth extinction. The prosecution concludes its case by arguing that mammoth extinction fits a clear pattern of human-caused extinctions that continues to the present day. The same combination of technological sophistication, rapid expansion, and overexploitation that characterized late Pleistocene human behavior continues to drive species extinctions in the modern world. Understanding human responsibility for mammoth extinction is essential for preventing similar tragedies from occurring with remaining megafauna populations. As the interrogation of humanity concludes, the evidence presents a compelling but not conclusive case for human responsibility in mammoth extinction. The correlation between human expansion and mammoth disappearance is undeniable, yet the complexity of late Pleistocene ecosystems leaves room for alternative explanations. The verdict against humanity remains uncertain, complicated by evidence that suggests multiple factors may have combined to create the perfect storm that eliminated one. The laboratory is eerily quiet except for the hum of refrigeration units that maintain ancient biological samples at temperatures that would have been familiar to the victims of this investigation. Dr. Ross McPhee adjusts his protective equipment as he prepares to examine evidence for the most elusive suspect in the mammoth extinction case, an invisible killer that leaves no fossils, no bones, and no direct traces of its presence. This suspect operates through biological warfare so sophisticated that it can eliminate entire species while leaving their predators and prey completely unaffected. Suspect number 3 enters the interrogation not as a single entity, but as a vast army of microscopic assassins that could have crossed continents in the guts of migrating humans and their domestic animals. Disease, specifically hyperdisease caused by pathogens introduced from Africa during human expansion, stands accused of conducting the most successful genocide in natural history through biological weapons that were invisible, silent, and absolutely lethal to populations that had no evolutionary experience with African pathogens. The motive is as old as evolution itself, the competitive advantage gained by eliminating competing species from shared habitats. When humans expanded out of Africa, carrying with them the pathogens that had co-evolved with human populations over millions of years, they inadvertently created biological weapons that could devastate naive animal populations that had never been exposed to African diseases. This was not intentional warfare, but accidental genocide, conducted by pathogens that found virgin populations with no immune defenses against invaders from another continent. The hyperdisease hypothesis, first proposed by Ross McPhee and Preston Marks in 1997, suggests that the rapid and synchronous extinctions that characterize the end of the Pleistocene could only have been caused by infectious diseases that spread faster than any human hunting campaign could have reached isolated populations. Traditional explanations for extinction require direct human contact with prey species, yet many mammoth populations disappeared from regions where archaeological evidence shows minimal or no human presence. The opportunity for pathogen transmission was created by the same global expansion of human populations that makes humanity a suspect in the mammoth extinction case. As humans spread across previously uninhabited continents, they carried with them not only their hunting technologies but also their internal parasites, their commensal bacteria, and the pathogens of their domestic animals. These biological hitchhikers were transported across oceanic and continental barriers that had previously prevented the global distribution of African diseases. The method of killing employed by this microscopic suspect is both elegant and terrifying in its effectiveness. Unlike hunting or climate change, which cause direct mortality through violence or resource depletion, disease can eliminate populations through subtle biological disruption that may not become apparent until entire ecosystems have collapsed. Pathogens can reduce reproductive success, impair immune function, or cause behavioral changes that make animals vulnerable to predation or environmental stress without causing immediate death. The forensic investigation of disease-mediated extinction faces unique challenges because pathogens rarely leave direct evidence in the fossil record. Soft tissues that might contain evidence of infection decompose rapidly under most conditions, while the microscopic organisms responsible for disease are too small and fragile to fossilize under normal circumstances. This creates a situation where the most devastating suspect in the case may also be the most difficult to detect using traditional forensic techniques. However, recent advances in ancient DNA technology have begun to reveal evidence of pathogen presence in archaeological specimens that previously would have been invisible to investigators. Genetic material from bacteria, viruses, and parasites can sometimes be recovered from bone samples, tooth enamel, and other mineralized tissues that preserve better than soft organs where pathogens typically reside. These molecular fossils provide the first direct evidence for pathogen involvement in ancient extinction events. The prosecution presents evidence from modern disease outbreaks that demonstrate the devastating effects that introduced pathogens can have on naive animal populations. The fungal disease that causes white nose syndrome has killed millions of bats across North America within just a few decades of its introduction from Europe. Chytrid fungus has driven numerous amphibian species to extinction across multiple continents through rapid population collapse that closely resembles the pattern seen in Pleistocene megafauna extinctions. The chronic wasting disease that affects deer and elk populations provides another modern example of how infectious agents can cause population collapse in large herbivores, similar to mammoths. This prion disease spreads through environmental contamination and can persist in soil for years, creating long-term population sinks that continue to affect animal populations long after the initial outbreak. The geographic spread of chronic wasting disease across North America demonstrates how quickly pathogens can disperse across continental scales. The prosecution argues that the rapid and synchronous nature of late Pleistocene extinctions is most consistent with disease-mediated mortality rather than hunting or climate change. Mathematical models of pathogen spread show that infectious diseases can eliminate populations across vast geographic areas within decades or centuries, much faster than human hunting campaigns could reach isolated populations or climate changes could affect local environments. The suspect's ability to cause selective extinction provides another line of evidence that supports the disease hypothesis. Unlike climate change, which should affect all species in an ecosystem equally, or hunting pressure, which should target the most visible and valuable prey species, disease can cause highly selective mortality that affects only those species that lack appropriate immune responses to specific pathogens. The prosecution introduces evidence that mammoth populations showed signs of genetic diversity loss during the period leading up to extinction, suggesting that populations were experiencing stress that reduced effective population sizes even before the final collapse. This pattern is consistent with endemic disease pressure that was slowly weakening mammoth populations by reducing reproductive success and increasing mortality rates without causing immediate extinction. The timing of mammoth extinction across different continents provides compelling circumstantial evidence for disease involvement. In most regions, mammoth populations collapsed within a few centuries of human arrival, regardless of local environmental conditions or the intensity of human hunting pressure. This synchrony is difficult to explain through hunting or climate change alone, but is consistent with the rapid spread of introduced pathogens following human colonization. However, the defense presents significant challenges to the disease hypothesis that raise reasonable doubt about pathogen involvement in mammoth extinction. The most damaging evidence against the disease suspect is the lack of direct pathogen remains in mammoth fossils, despite extensive searching by multiple research teams using state-of-the-art molecular techniques. The defense argues that if disease was the primary cause of mammoth extinction, then evidence of pathogen infection should be detectable in at least some of the thousands of mammoth specimens that have been recovered from permafrost deposits. The excellent preservation conditions in Arctic environments should have maintained pathogen DNA or protein signatures if disease was widespread in mammoth populations during the extinction period. Furthermore, the defense points out that disease outbreaks typically affect multiple species within an ecosystem rather than targeting a single species for extinction. If introduced pathogens were responsible for mammoth extinction, then other large herbivores that shared mammoth habitats should also have been affected. The survival of bison, caribou, musk oxen, and other species that lived alongside mammoths suggests that pathogen pressure was not sufficient to cause ecosystem-wide collapse. The defense introduces expert testimony from epidemiologists who argue that the population structure of mammoth herds would have provided natural protection against pandemic disease spread. Mammoth populations were dispersed across vast territories in small family groups that had limited contact with each other, creating natural fire breaks that should have prevented rapid disease transmission across entire populations. The prosecution counters with evidence from modern disease outbreaks showing that social behaviors that normally provide protection against disease spread can become vectors for transmission when populations encounter novel pathogens. The strong family bonds and social cooperation that characterized mammoth herds could have facilitated disease transmission if infected individuals were cared for by healthy family members who subsequently became infected themselves. The prosecution presents evidence that some diseases can persist in the environment for extended periods, creating contaminated landscapes that continue to infect new individuals long after initial outbreaks have ended. Anthrax spores can remain viable in soil for decades, while some viral pathogens can persist in water sources or on vegetation for months or years. These environmental reservoirs could have maintained infection pressure on mammoth populations, even in regions where direct contact with infected individuals was minimal. The defense argues that the proposed African origin of mammoth pathogens is inconsistent with the biogeographic patterns of disease distribution. Most infectious diseases show strong geographic clustering that reflects their co-evolutionary history with specific host populations. Diseases that evolved in tropical African environments would be unlikely to survive transmission to Arctic environments where mammoth populations lived. However, the prosecution responds with evidence from modern research showing that many diseases can be transmitted through environmental contamination rather than direct contact between individuals. Mammoth populations that use the same water sources, migration routes, or feeding areas as infected humans or domestic animals could have been exposed to pathogens through environmental vectors even without direct interspecies contact. The prosecution introduces evidence from ancient DNA studies showing that some mammoth populations experienced rapid declines in genetic diversity that preceded morphological evidence of extinction by several thousand years. This pattern suggests that populations were experiencing demographic stress that reduced effective population sizes, creating conditions that would have made them more vulnerable to disease outbreaks and other mortality factors. The defense argues that genetic diversity loss could have been caused by factors other than disease, including habitat fragmentation due to climate change or hunting pressure that reduced population sizes below levels necessary to maintain genetic health. The defense points out that small populations are vulnerable to extinction from multiple causes, making it difficult to identify disease as the specific factor responsible for final population collapse. The prosecution presents its final evidence in the form of mathematical models showing that disease outbreaks can cause population extinctions even when mortality rates are relatively low, provided that the disease affects reproductive success or juvenile survival. Diseases that reduce mammoth reproductive success by even modest amounts could have caused population decline that would accelerate over successive generations until extinction became inevitable. The prosecution argues that the invisible nature of disease makes it the perfect suspect for a crime that left minimal direct evidence. Unlike hunting, which leaves cut marks on bones and spear points in skeletons, or climate change, which leaves clear environmental signatures in geological records, disease can eliminate entire species while leaving only subtle genetic and demographic signatures that are difficult to detect using traditional forensic techniques. The defense makes its closing argument by emphasizing the lack of direct pathogen evidence in mammoth remains, despite extensive searching using the most advanced molecular techniques available. The defense argues that extraordinary claims require extraordinary evidence, and that the disease hypothesis remains speculative until direct evidence of pathogen infection can be demonstrated in mammoth specimens. However, the prosecution concludes by arguing that the absence of evidence is not evidence of absence, particularly when dealing with biological materials that are inherently unlikely to preserve over geological timescales. The prosecution points out that most modern disease outbreaks would leave no detectable trace in the fossil record if they occurred thousands of years ago. Yet their devastating effects on contemporary animal populations demonstrate the potential for disease to cause rapid species extinction. The case against disease as the primary cause of mammoth extinction presents unique challenges for forensic analysis because it requires proving the existence of evidence that may no longer exist. The circumstantial evidence for pathogen involvement is compelling, particularly the rapid and synchronous nature of extinctions across multiple continents. Yet the lack of direct pathogen remains in mammoth fossils creates reasonable doubt about the role of disease in driving mammoth populations to extinction. As the interrogation of the disease suspect concludes, investigators are left with a compelling theory that explains many aspects of mammoth extinction while remaining fundamentally unprovable using current forensic techniques. The invisible killer remains at large, neither convicted nor exonerated, leaving open the possibility that the greatest extinction event in recent natural history may have been caused by microscopic assassins that have long since vanished from the crime scene. The witness stand is occupied not by a person, but by a single molar tooth, dark with age and worn smooth by decades of grinding tundra grass. This tooth belongs to one of the last mammoths ever to walk the earth, a member of the final population that survived on Wrangel Island until just 4,000 years ago, when the pyramids of Egypt were already ancient monuments. Dr. Love Delaine adjusts his DNA sequencing equipment as he prepares to extract testimony from the genetic material preserved within this tooth. Testimony that will reveal the final chapter in the mammoth extinction case and provide crucial evidence about how the last survivors met their fate. The final witnesses in this extinction investigation are not ancient bones preserved in museums, but living genetic sequences that can still be read from the DNA of the last mammoth populations to survive the Great Dying. These final populations represent crucial evidence because they survived thousands of years longer than their mainland relatives, providing natural experiments that can help determine which of our suspects was ultimately responsible for driving mammoths to extinction. Wrangel Island sits like a frozen courtroom in the Arctic Ocean, a small piece of land that became the final refuge for a population of mammoths that had escaped whatever catastrophe eliminated their relatives across the rest of the world. This island, roughly the size of Crete, supported a stable population of between 500 and 1,000 mammoths for over 6,000 years after mainland extinctions had occurred, creating a unique opportunity to observe how mammoth populations behaved when protected from the primary suspects in the extinction case. The testimony from Wrangel Island challenges many of the assumptions that have dominated the mammoth extinction investigation. If climate change was the primary cause of mammoth extinction, then the island population should have disappeared at the same time as mainland herds, since Wrangel Island experienced the same warming trends that affected the rest of the Arctic. If human hunting was responsible for mammoth extinction, then the island population should have survived indefinitely, Protected by oceanic barriers that prevented human contact until much later in their history, the genetic evidence extracted from Wrangel Island mammoth remains tells a story that is both tragic and scientifically illuminating. DNA analysis reveals that the island population was founded by a small group of mammoths that became isolated when rising sea levels separated Wrangel Island from the Siberian mainland approximately 12,000 years ago. This founding population probably consisted of fewer than 50 individuals, creating an immediate genetic bottleneck that would influence the fate of all subsequent generations. The molecular autopsy of the Wrangel Island population reveals the slow-motion genetic catastrophe that unfolded over thousands of years as the isolated mammoth community struggled with the consequences of inbreeding and genetic drift. Each generation saw further erosion of the genetic diversity that had sustained mammoth populations for hundreds of thousands of years, as random mutations accumulated and beneficial genes were lost through the lottery of reproduction in a population too small to maintain evolutionary stability. The forensic geneticists who have analyzed DNA from Wrangel Island mammoths describe finding genetic signatures that resemble those seen in severely inbred domestic animals or endangered species that have passed through extreme population bottlenecks. Levels of heterozygosity, the genetic diversity that provides populations with evolutionary flexibility, declined steadily throughout the island's mammoth history until the final generations showed genetic uniformity that approached that of identical twins. However, the remarkable fact about the Wrangel Island population is not that they eventually succumbed to genetic problems, but that they survived for over 6,000 years despite severe inbreeding and genetic deterioration. This extended survival suggests that mammoth populations possessed remarkable resilience that enabled them to persist under conditions that would have quickly eliminated most other large mammal species. The island mammoths were not thriving, but they were surviving, maintaining stable population numbers and continuing to reproduce despite accumulating genetic damage. The environmental conditions on Wrangel Island during the period of mammoth survival provide crucial evidence about the habitat requirements and environmental tolerances of late surviving mammoth populations. Pollen records and other paleoenvironmental data indicate that the island maintained patches of mammoth steppe vegetation long after similar habitats had disappeared from mainland areas, suggesting that suitable mammoth habitat persisted in some locations despite global climate change. The archaeological evidence from Wrangel Island reveals that humans did not reach the island until approximately 3,700 years ago, meaning that the mammoth population survived for over 8,000 years without any human contact. This isolation experiment demonstrates that mammoth populations could persist for millennia without human hunting pressure, provided that suitable habitat was available and population sizes remained above critical thresholds for genetic viability. The testimony from other island mammoth populations provides additional evidence about the factors that determined mammoth survival or extinction. The Channel Islands off the coast of California supported populations of dwarf mammoths that survived until approximately 13,000 years ago, while Mediterranean islands, including Cyprus, Crete, and Sicily, had their own dwarf mammoth populations that persisted until various times during the late Pleistocene and early Holocene. The phenomenon of island dwarfism exhibited by these isolated mammoth populations provides important insights into mammoth evolutionary potential and adaptability. Within relatively few generations, island mammoth populations evolved smaller body sizes that were better suited to the limited resources available on their island homes. The Channel Island mammoths, known as Mammothus exilis, stood only about 1.8 meters tall at the shoulder, roughly half the size of their mainland relatives. This rapid evolutionary response to island conditions demonstrates that mammoth populations retain the genetic flexibility necessary for adaptation to new environments, contradicting arguments that mammoths were evolutionary dead ends that were inevitably doomed to extinction. The successful adaptation of multiple mammoth populations to island environments shows that these animals could modify their biology in response to environmental challenges when given sufficient time and appropriate conditions. The chronology of island mammoth extinctions provides a complex pattern that challenges simple explanations for mammoth disappearance. Some island populations disappeared at roughly the same time as mainland extinctions, while others persisted for thousands of years beyond the mainland collapse. The Wrangel Island population survived the longest, finally disappearing around 4,000 years ago during a period when human populations were present on the island, but environmental conditions remained relatively stable. The forensic analysis of the final Wrangel Island extinctions reveals genetic evidence for the process that ultimately eliminated this last mammoth population. DNA sequences from the latest specimens show extreme genetic uniformity and high frequencies of deleterious mutations that would have reduced individual fitness and population viability. The final generations appear to have suffered from genetic meltdown, a process where accumulating mutations reduce population fitness to levels that cannot sustain reproduction. However, the timing of the final Wrangel Island extinction remains controversial, with some evidence suggesting that human arrival on the island may have provided the final push that eliminated a mammoth population already weakened by genetic problems. Archaeological evidence shows that humans were present on Wrangel Island during the final centuries of mammoth survival, creating the possibility that hunting pressure or other human activities contributed to the final extinction event. The defense argues that the Wrangel Island evidence exonerates climate change as the primary cause of mammoth extinction, since the island population survived for thousands of years after mainland climate changes had supposedly eliminated mammoth habitat. The persistence of mammoth steppe vegetation on Wrangel Island long after its disappearance from mainland areas suggests that suitable mammoth habitat could have persisted in other refugial areas if climate change alone was responsible for mainland extinctions. The prosecution counters that the Wrangel Island population represents a special case that does not invalidate the climate change hypothesis for mainland extinctions. The island's maritime climate and isolation from continental weather patterns may have buffered it from the most severe effects of post-glacial warming, allowing mammoth habitat to persist longer than on the mainland where continental climate changes were more extreme. The human hunting hypothesis faces challenges from the Wrangel Island evidence, since the mammoth population survived for thousands of years without human contact and only disappeared after humans arrived on the island. However, the prosecution argues that the timing of human arrival and mammoth extinction on Wrangel Island actually supports the hunting hypothesis by demonstrating that mammoth populations remained stable until human contact occurred. The disease hypothesis gains some support from the Wrangel Island evidence, since the final extinction occurred after human arrival on the island, and therefore after the first opportunity for pathogen transmission from human populations to mammoths. However, the genetic evidence for inbreeding depression and genetic meltdown provides an alternative explanation for the final extinction that does not require disease involvement. The St. Paul Island mammoth population provides additional testimony about island mammoth extinctions. This population, isolated on a small island in the Bering Sea, survived until approximately 5,600 years ago before disappearing in what appears to have been a relatively rapid extinction event. Genetic analysis of St. Paul mammoth remains reveals patterns similar to those seen on Wrangel Island, with evidence for genetic bottlenecks and inbreeding depression in the final populations. The environmental history of St. Paul Island during the period of mammoth survival shows that the island experienced gradual environmental changes that reduced the amount of suitable mammoth habitat over time. Lake sediment cores reveal evidence for vegetation changes and possible freshwater shortages that would have stressed the mammoth population during its final centuries. The combination of genetic problems and environmental degradation appears to have created conditions that made the small mammoth population unsustainable. The Mediterranean dwarf mammoth populations provide yet another perspective on island mammoth extinctions, with different species showing various patterns of survival and disappearance that span tens of thousands of years. The Sicilian dwarf mammoth, Mammoths falconeri, survived until approximately 17,000 years ago, while the Cypriot dwarf mammoth persisted until around 11,000 years ago. These varying extinction times suggest that local factors rather than global phenomena were responsible for eliminating different island populations. The prosecution presents evidence that the staggered timing of island mammoth extinctions supports a complex multi-causal explanation for mammoth disappearance rather than a single global catastrophe. Each island population faced its own combination of environmental challenges, genetic problems, and human contact that determined its ultimate fate. This pattern suggests that mainland mammoth extinctions resulted from similar combinations of factors that varied by region and population. The defense argues that the success of island mammoth populations in surviving mainland extinction events demonstrates that mammoths possess the biological capacity to persist through the environmental changes that occurred at the end of the Ice Age. The fact that multiple island populations survived for thousands of years beyond mainland extinctions shows that suitable conditions for mammoth survival continued to exist in some locations despite global climate change. The genetic testimony from island mammoth populations reveals important information about the minimum population sizes necessary for long-term mammoth survival. Population genetic models based on island mammoth DNA suggest that populations of several hundred individuals can persist for thousands of years despite inbreeding, provided that environmental conditions remain suitable and external mortality factors are minimized. However, the same genetic evidence demonstrates that populations below certain critical thresholds will eventually succumb to genetic problems regardless of environmental conditions. The Wrangel Island population appears to have hovered near this threshold for most of its existence, surviving much longer than expected for such a small and isolated group, but ultimately falling victim to the genetic deterioration that inevitably affects severely bottlenecked populations. The final testimony from the last mammoth witnesses reveals a complex picture of extinction that defies simple explanations and single causes. The survival of island populations for thousands of years beyond mainland extinctions demonstrates that mammoth disappearance was not the result of a single global catastrophe, but rather a series of regional events that eliminated populations under different combinations of circumstances. The Wrangel Island mammoths, as the final survivors of their species, provide crucial evidence that mammoths possessed remarkable resilience and could adapt to changing conditions when given appropriate opportunities. Their eventual extinction appears to have resulted from the intersection of genetic problems caused by small population size and environmental or human factors that provided the final stress that eliminated a population already weakened by thousands of years of inbreeding. The last witnesses in the mammoth extinction case tell a story of gradual decline rather than sudden catastrophe, of populations that fought for survival against mounting genetic and environmental pressures before finally succumbing to forces that had been building for millennia. Their testimony suggests that mammoth extinction was not inevitable, but rather the result of circumstances that prevented populations from maintaining the genetic diversity and environmental stability necessary for long-term survival. As the final witnesses conclude their genetic testimony, the evidence points toward a complex intersection of factors that combine to eliminate mammoth populations across different times and places. The last survivors on remote islands demonstrate both the potential for mammoth persistence under favorable conditions and the ultimate vulnerability of small populations to genetic and environmental pressures that gradually erode their capacity for survival. The laboratory resembles a high-tech forensics facility where the most advanced crime-solving technologies in the world are being applied to evidence that is thousands of years old. Dr. Beth Shapiro removes a tiny sample of bone powder from a mammoth femur as she prepares to conduct the most sophisticated genetic analysis ever attempted on extinct species DNA. This is not just another scientific experiment. This is the final forensic examination that will either solve the mammoth extinction case or leave it forever unsolved, using molecular detective work that can read the story written in genetic code preserved within ancient bones. The DNA laboratory has become the ultimate courtroom where genetic evidence provides testimony that cannot lie, cannot be intimidated, and cannot change its story under cross-examination. The molecules extracted from mammoth bones carry within their chemical structure the complete history of mammoth populations, their movements across continents, their responses to environmental change, and the gradual or sudden processes that led to their extinction. This genetic testimony represents the most reliable witness in the entire investigation. The ancient DNA revolution that began in the 1980s has transformed paleontology from a science that could only describe what extinct animals look like into a discipline that can reconstruct how they lived, how they died, and why they disappeared. The genetic material preserved in mammoth bones provides access to biological information that was completely inaccessible to previous generations of researchers who had to rely on skeletal morphology and archaeological context to understand prehistoric life. The technical challenges involved in extracting and analyzing ancient DNA rival those faced by forensic scientists working with degraded evidence from modern crime scenes. DNA molecules begin breaking down immediately after an organism dies, fragmenting into smaller and smaller pieces that become increasingly difficult to detect and sequence. The preservation of readable genetic material for tens of thousands of years requires exceptional environmental conditions that prevent bacterial decay and chemical degradation. The permafrost environments where most mammoth DNA has been recovered create natural freezers that can preserve genetic material with unprecedented fidelity. The constantly frozen conditions prevent bacterial activity that would normally consume organic molecules within years or decades of death, while the absence of liquid water prevents chemical reactions that would break down DNA structure. Some mammoth specimens recovered from Siberian permafrost contain DNA that is so well preserved it can be sequenced using the same techniques applied to modern biological samples. The genetic analysis begins with the painstaking process of extracting DNA from bone samples that must be handled with extreme care to prevent contamination with modern genetic material. Ancient DNA laboratories operate under sterile conditions that exceed those found in hospital operating rooms, with researchers wearing full protective equipment and following protocols designed to prevent even microscopic amounts of modern DNA from contaminating precious ancient samples. The DNA extraction process reveals the first clues about mammoth population dynamics during the period leading up to extinction. Genetic material from mammoth bones of different ages provides a chronological record of genetic diversity changes that can be used to reconstruct population sizes, migration patterns, and breeding success over thousands of years. This molecular timeline provides evidence that cannot be obtained through any other forensic technique. The initial results from genetic analysis immediately challenged several assumptions about mammoth extinction that have dominated scientific thinking for decades. Rather than revealing a single mammoth species that went extinct simultaneously across the globe, DNA analysis shows that mammoths consisted of multiple genetically distinct populations that had been evolving independently for hundreds of thousands of years before extinction occurred. The genetic evidence reveals that woolly mammoths and Columbian mammoths, previously thought to be separate species, were actually part of a continuous population that exchanged genetic material across the Bering Land Bridge throughout much of their evolutionary history. This discovery indicates that mammoth populations were more connected and genetically diverse than previously suspected, making their extinction more puzzling rather than less so. The molecular analysis uncovers evidence for multiple mammoth lineages that had adapted to different environmental conditions across the vast northern hemisphere range. Some genetic lineages show adaptations for life in forested environments, while others retain signatures of adaptation to grassland habitats. This genetic diversity should have provided insurance against extinction by ensuring that some populations could survive environmental changes that eliminated others. The DNA testimony reveals dramatic population fluctuations that occurred throughout mammoth evolutionary history, with genetic bottlenecks and population expansions that correlate with major climate changes over hundreds of thousands of years. These genetic patterns show that mammoth populations had successfully navigated previous environmental crises that were similar in magnitude to the changes that occurred at the end of the Ice Age. However, the genetic analysis also reveals ominous signs that mammoth populations were experiencing unprecedented stress during the final millennia before extinction. DNA samples from the latest mammoth populations show evidence of declining genetic diversity that suggests population sizes were shrinking rapidly during the period when climate change, human expansion, and possible disease introduction were all occurring simultaneously. The forensic geneticists discover evidence for population fragmentation that occurred during the late Pleistocene, with previously connected mammoth populations becoming isolated from each other as environmental changes disrupted traditional migration routes. This fragmentation would have prevented gene flow between populations that had maintained genetic connectivity for hundreds of thousands of years, reducing the overall resilience of the species to environmental stress. The genetic evidence for interbreeding between different mammoth lineages during the extinction period suggests that populations were being forced into contact in refugial areas as their traditional habitats disappeared. This mixing of previously separated genetic lineages created hybrid populations that may have been less well adapted to local environmental conditions than their parental species. The molecular analysis reveals evidence for rapid evolutionary change In mammoth populations during the final phases of their existence, with genetic signatures indicating selection for smaller body size, modified metabolic rates, and other adaptations that would have been advantageous in changing environmental conditions. These genetic changes demonstrate that mammoth populations were responding to environmental pressure through evolutionary adaptation, rather than simply declining toward extinction. However, the DNA evidence also shows that the rate of environmental change during the late Pleistocene exceeded the rate at which mammoth populations could adapt through genetic evolution. Mathematical models based on mammoth generation times and observed rates of genetic change indicate that adaptive evolution could not have kept pace with the environmental changes that characterized the end of the Ice Age. The genetic analysis provides crucial evidence about the role of inbreeding depression in mammoth extinction by revealing patterns of genetic diversity loss that preceded morphological evidence of population decline. DNA from mammoth populations shows increasing homozygosity and accumulation of deleterious mutations during the final millennia before extinction, indicating that genetic problems were contributing to population stress even before environmental or human factors delivered final blows. The forensic geneticists discover evidence for genetic rescue attempts that occurred as different mammoth populations came into contact during the extinction period. Gene flow between previously isolated populations temporarily increased genetic diversity in some groups, but this genetic rescue was apparently insufficient to prevent ultimate extinction as environmental and other pressures continued to intensify. The DNA testimony reveals important information about mammoth population structure and social organization that provides clues about vulnerability to different extinction causes. Genetic analysis of family relationships within mammoth herds shows that social groups consisted of closely related females and their offspring, creating population structures that would have been vulnerable to selective hunting of reproductive females, but resistant to random environmental mortality. The DNA evidence provides insights into mammoth reproductive biology that are relevant to understanding their vulnerability to hunting pressure and environmental stress. Genetic analysis reveals that mammoth reproductive rates were similar to those of modern elephants, with long generation times and low reproductive output that made populations vulnerable to any factor that increased adult mortality or reduced reproductive success. The molecular analysis discovers evidence for sex-biased mortality in late mammoth populations, with DNA from the extinction period showing skewed sex ratios that suggest differential survival between males and females. This pattern could indicate selective hunting pressure that preferentially targeted one sex, or environmental factors that affected male and female mammoths' survival differently. The genetic testimony reveals evidence for population connectivity between mammoth populations on different continents that persisted until very late in mammoth evolutionary history. DNA analysis shows gene flow between Eurasian and North American mammoth populations continued until within a few thousand years of extinction, indicating that genetic isolation was not a factor in mammoth disappearance until very late in the process. However, the same genetic evidence shows that this intercontinental gene flow ceased abruptly during the final millennia before extinction, suggesting that whatever factors caused mammoth extinction also disrupted the long-distance movements that had maintained genetic connectivity between continents throughout mammoth evolutionary history. The forensic genetic analysis provides the most detailed reconstruction ever attempted of mammoth population dynamics during the extinction period. Mathematical models based on genetic data suggest that mammoth populations declined gradually for several millennia before collapsing rapidly during the final centuries before extinction, indicating that multiple factors were probably involved in creating conditions that ultimately proved unsustainable. The DNA evidence reveals that different mammoth populations experienced extinction at different times and through different processes, with some showing evidence of gradual decline over thousands of years, while others appeared to have collapsed rapidly within a few centuries. This pattern suggests that mammoth extinction was not a single event, but rather a series of regional collapses that occurred under different combinations of environmental, human, and genetic factors. The genetic analysis provides evidence for mammoth population recovery attempts that occurred during temporary environmental improvements within the overall extinction period. DNA samples from some regions show evidence of population expansion and genetic diversification during brief periods when environmental conditions became more favorable. demonstrating that mammoth populations retain the capacity for recovery even during the overall decline toward extinction. However, the same genetic evidence shows that these recovery attempts were ultimately unsuccessful, with population crashes that eliminated any gains made during favorable periods. The failure of mammoth populations to establish sustainable recovery, despite temporary improvements in conditions, suggests that multiple limiting factors were operating simultaneously to prevent long-term population stability. The final genetic testimony reveals that mammoth extinction was ultimately the result of demographic stochasticity in populations that had been reduced to critically small sizes by the interaction of environmental change, human pressure, and genetic problems. Once mammoth populations fell below certain threshold sizes, random fluctuations in birth and death rates created positive feedback loops that drove populations toward extinction, regardless of environmental conditions or human activities. The DNA evidence demonstrates that mammoth populations possessed remarkable resilience that enabled them to survive environmental challenges, hunting pressure, and genetic problems that would have eliminated most other large mammal species. The fact that mammoths ultimately succumbed to extinction despite this resilience indicates that the combination of factors they faced during the late Pleistocene was unprecedented in its severity and synchrony. As the genetic investigation concludes, the DNA evidence provides the most comprehensive view yet available of the complex processes that led to mammoth extinction. The molecular testimony reveals a species that fought for survival against mounting environmental, human, and genetic pressures before finally succumbing to a perfect storm of circumstances that overwhelmed even the jury deliberation room is silent, except for the quiet hum of climate-controlled storage units containing the evidence that has been presented over months of investigation into the greatest extinction case in natural history. 3 sets of evidence files sit on the table before the panel of expert witnesses who must now weigh the testimony of permafrost-preserved DNA, isotope signatures from ancient bones, archaeological kill sites, and climate records spanning millennia. This is not a trial where guilt or innocence can be determined by a single smoking gun or eyewitness account. This is a complex investigation where multiple suspects had motive, opportunity, and method to commit the crime that eliminated 5 million woolly mammoths from the face of the earth within the span of just a few thousand years. Dr. Daniel Fisher adjusts his reading glasses as he reviews the evidence summary that will determine how this case is resolved. The forensic team has spent years analyzing every piece of available evidence using techniques that would have been pure science fiction when the crime was committed 10,000 years ago. Ancient DNA sequencing, isotope mass spectrometry, radiocarbon dating, and computer modeling have all been deployed to reconstruct the final chapters in mammoth evolutionary history with unprecedented precision and detail. The standard of proof required in this extinction investigation goes beyond reasonable doubt to approach scientific certainty. The verdict must be based on evidence that can withstand peer review, replication, and the scrutiny of researchers who will spend decades testing and retesting every conclusion. This is not merely an academic exercise, but a determination that will influence how humanity responds to current extinction crises and future conservation challenges. The first suspect to face final judgment is climate change, the massive environmental transformation that ended the Ice Age and created the modern world. The prosecution has presented compelling evidence that rapid warming between 15 and 10,000 years ago eliminated 90% of mammoth habitat across the northern hemisphere. The mammoth steppe ecosystem that had supported millions of large herbivores was replaced by forests, wetlands, and tundra that could not sustain the same biomass of megafauna. The evidence for climate change culpability is written in ice cores, lake sediments, and pollen records that document the most rapid and extensive environmental transformation since the extinction of the dinosaurs. Temperature increases of 5 to 10 degrees Celsius occurred within centuries rather than millennia, creating environmental changes that outpace the ability of mammoth populations to adapt through migration or behavioral modification. The timing of these environmental changes correlates precisely with the first evidence of mammoth population decline across multiple continents. The prosecution presents isotope evidence from mammoth bones showing that diet quality deteriorated markedly during the final millennia before extinction. Mammoth teeth from the latest populations show wear patterns indicating that animals were forced to consume increasingly fibrous and less nutritious vegetation as their preferred food sources disappeared. Stomach contents from mummified mammoth carcasses reveal that final populations were subsisting on low-quality browse rather than the nutrient-rich grasses that had sustained their ancestors. The geographic pattern of mammoth extinction provides additional evidence for climate change involvement. Northern populations that experienced the most severe warming disappeared first, while southern populations persisted longer in regions where environmental changes were less extreme. Island populations survived longest of all in maritime climates that were buffered from continental temperature extremes, demonstrating that mammoth survival was directly linked to environmental stability. However, the defense presents significant challenges to the climate change hypothesis that create reasonable doubt about environmental factors as the primary cause of mammoth extinction. The most damaging evidence against climate change culpability is the fact that mammoth populations had successfully survived multiple previous episodes of rapid warming that were similar in magnitude and rate to the changes that occurred at the end of the Ice Age. Paleoclimatic records show that mammoth ancestors survived at least 20 major climate transitions during their 400,000-year evolutionary history, including warming episodes that eliminated their preferred habitats for thousands of years at a time. Each time, mammoth populations had recovered and expanded when favorable conditions returned. The defense argues that there is no evidence that late Pleistocene climate change was more severe or rapid than previous environmental transitions that mammoth populations had survived. Furthermore, the defense points out that many mammoth populations disappeared from regions where suitable habitat continued to exist long after mainland extinctions had occurred. Pollen records from some areas show that mammoth steppe vegetation persisted for thousands of years after local mammoth extinctions, indicating that habitat availability was not the limiting factor preventing mammoth survival in these regions. The prosecution counters with evidence that late Pleistocene climate change was qualitatively different from previous environmental transitions because it coincided with other mortality factors that had not been present during earlier climate cycles. The prosecution argues that climate change alone might not have caused mammoth extinction, but that environmental stress created conditions that made mammoth populations vulnerable to hunting pressure and disease outbreaks that would not have been lethal under normal circumstances. The second suspect to face final judgment is Homo sapiens, the globally expanding population of technologically sophisticated hunters who had the motive, opportunity, and method to drive mammoth populations to extinction through systematic overhunting. The prosecution presents archaeological evidence for direct human predation on mammoth populations, including kill sites with butchering marks on mammoth bones and spear points embedded in mammoth skeletons. The human hunting hypothesis gains support from the correlation between human expansion and megafauna extinctions on multiple continents. In North America, Clovis hunters arrived approximately 13,000 years ago, and mammoth populations collapsed within a few centuries of human contact. In Australia, human arrival 45,000 years ago coincided with the extinction of giant marsupials, including diprotodonts and giant kangaroos. In Madagascar, human colonization 2,000 years ago was followed by the rapid elimination of elephant birds and giant lemurs. The prosecution argues that humans represented an unprecedented predation pressure that mammoth populations had no evolutionary experience to resist. Unlike other predators that had co-evolved with mammoth populations over hundreds of thousands of years, humans used cooperative hunting strategies, advanced weapons technology, and fire manipulation that could overwhelm even large, dangerous prey species that had few natural enemies. Mathematical models presented by the prosecution demonstrate that sustainable human hunting could have eliminated mammoth populations within centuries if hunters preferentially targeted reproductive females and juvenile animals. The slow reproductive rate of mammoth populations made them particularly vulnerable to any increase in adult mortality, while their social behavior patterns created opportunities for hunters to kill multiple animals from single herds. However, the defense presents compelling evidence that challenges the human hunting hypothesis and raises serious doubts about the ability of small human populations to drive millions of mammoths to extinction. Archaeological evidence shows that human populations during the late Pleistocene were tiny compared to mammoth populations, with perhaps tens of thousands of hunters across the entire northern hemisphere, facing millions of prey animals distributed across 13 million square kilometers. The defense argues that human hunting technology, while advanced for its time, was still primitive compared to modern weapons and would have been insufficient to kill large numbers of mammoth efficiently. Experimental archaeology shows that killing adult mammoths with spears and atlatls was dangerous, difficult, and often unsuccessful, even for experienced hunters working in coordinated groups. Furthermore, the defense points out that humans and mammoths coexisted for thousands of years in some regions without causing mammoth extinction. In Europe, humans and mammoths shared the landscape for over 30,000 years before mammoth populations collapsed, suggesting that sustainable coexistence was possible and that hunting pressure alone was not sufficient to drive mammoth populations to extinction. The prosecution responds with evidence that late Pleistocene human populations were qualitatively different from earlier human groups in their hunting technology, social organization, and environmental impact. Clovis hunters in North America and their contemporaries in other regions possessed fluted projectile points, atlatl throwing systems, and coordinated hunting strategies that were far more effective than the simple spears used by earlier human populations. The prosecution concludes its case by arguing that the evidence supports a verdict of guilty for all three suspects, with mammoth extinction resulting from the synergistic interaction of climate change, human hunting, and possible disease introduction. The prosecution contends that no single factor was sufficient to cause mammoth extinction, but that the combination of multiple stressors created conditions that overwhelmed mammoth population resilience and drove the species to extinction. The prosecution presents its final argument using the analogy of a perfect storm, where individual weather systems that would be manageable separately combine to create devastating conditions that exceed the ability of even experienced mariners to survive. Climate change weakened mammoth populations by reducing habitat quality and availability. Human hunting added direct mortality pressure that particularly affected reproductive females and social groups. Possible disease introduction created additional mortality that could have affected even healthy individuals in stable environments. The defense makes its closing argument by emphasizing that extraordinary claims require extraordinary evidence, and that the prosecution has failed to meet the burden of proof required to convict any suspect of primary responsibility for mammoth extinction. The defense argues that climate change was not more severe than previous environmental transitions that mammoth populations had survived. Human populations were too small and hunting technology too primitive to eliminate millions of mammoths across vast geographic areas. Disease remains purely speculative in the absence of any direct pathogen evidence. The defense concludes by arguing that mammoth extinction may have resulted from factors that are not represented among the current suspects, including cosmic impacts, volcanic eruptions, genetic problems, or ecological cascades that have not been identified by current research. The defense contends that the case should remain open pending the discovery of new evidence that can definitively identify the cause of mammoth extinction. After reviewing all evidence and arguments, the expert panel reaches its verdict through careful consideration of the scientific standards that must guide any conclusion about prehistoric events. The verdict must be based on evidence that can be independently verified and replicated, while acknowledging the limitations inherent in reconstructing events that occurred thousands of years ago using fragmentary evidence. The panel finds climate change guilty of contributing to mammoth extinction through habitat destruction and environmental stress that reduced mammoth population viability during the late Pleistocene. The evidence for rapid environmental change during the extinction period is overwhelming and well documented through multiple independent lines of evidence. However, the panel finds that climate change alone was not sufficient to cause mammoth extinction, since previous environmental transitions of similar magnitude had been survived by mammoth populations. The panel finds humans guilty of contributing to mammoth extinction through hunting pressure that added significant mortality to mammoth populations already stressed by environmental change. The correlation between human expansion and megafauna extinctions on multiple continents provides compelling evidence for human involvement, while archaeological evidence demonstrates direct predation on mammoth populations. However, the panel finds that human hunting alone was not sufficient to explain the rapid and synchronous extinctions that characterized the end of the Mammoth era. The panel finds the disease hypothesis not proven due to insufficient evidence for pathogen involvement in mammoth extinction. While the circumstantial evidence for disease involvement is intriguing and the potential for introduced pathogens to cause rapid population collapse is well established, the complete absence of direct pathogen evidence in mammoth specimens creates reasonable doubt about disease as a significant factor in mammoth extinction. The final verdict is that mammoth extinction resulted from the synergistic interaction of climate change and human hunting, with each factor amplifying the effects of the other to create conditions that exceeded mammoth population resilience. Climate change reduced habitat quality and availability, fragmenting mammoth populations and reducing carrying capacity. Human hunting added direct mortality pressure that was particularly devastating to populations already stressed by environmental change. The panel concludes that neither factor alone would have been sufficient to cause mammoth extinction, but that their combined effect created a mortality vice that squeezed mammoth populations from multiple directions simultaneously. Environmental stress reduced mammoth reproductive success and survival rates, while hunting pressure increased adult mortality beyond sustainable levels. The result was a demographic spiral that drove mammoth populations below minimum viable thresholds and ultimately to extinction. This verdict has profound implications for understanding current extinction crises and developing conservation strategies for endangered species. The Mammoth case demonstrates that extinction can result from the interaction of multiple factors that individually might be manageable, but collectively create unsustainable conditions for population persistence. Modern conservation efforts must consider not just single threats, but the cumulative effects of multiple stressors acting on vulnerable populations. The Mammoth extinction case also illustrates the importance of population resilience in determining species survival under changing conditions. Mammoth populations possessed remarkable adaptive capacity that enabled them to survive hundreds of thousands of years of environmental change and predation pressure. Their ultimate extinction demonstrates that even highly resilient species can be overwhelmed when faced with unprecedented combinations of mortality factors. The verdict in the Mammoth extinction case serves as both a historical resolution and a contemporary warning. The same factors that drove mammoths to extinction 10,000 years ago continue to threaten large mammal populations today. Climate change is altering habitats and environmental conditions at unprecedented rates, while human activities create direct and indirect mortality pressures on vulnerable species. The Mammoth case provides a cautionary tale about the consequences of inaction when multiple threats converge on populations that lack the resilience to withstand cumulative impacts. As the investigation concludes, the evidence has been weighed and the verdict rendered, but the lessons from the Mammoth extinction case remain relevant to current conservation challenges. The case is solved, but the work of preventing future extinctions has only just begun. The laboratory of Dr. George Church at Harvard Medical School resembles a combination of high-tech forensics facility and futuristic resurrection chamber, where the most advanced genetic engineering technologies ever developed are being applied to the unprecedented challenge of bringing back the dead. Banks of DNA sequencing machines, gene editing equipment, and cell culture incubators represent humanity's attempt to make amends for the Mammoth extinction case through the ultimate form of restorative justice, returning the victims to life after 10,000 years in the grave. The concept of de-extinction represents a radical departure from traditional conservation approaches that focus on protecting species before they disappear forever. Instead of accepting extinction as permanent and irreversible, the de-extinction movement proposes using advances in genetic technology to resurrect lost species and restore ecosystems that have been simplified by human activities. The mammoth represents the flagship species for this biological resurrection project, chosen because of extensive genetic information available from well-preserved specimens and the potential ecological benefits of returning large herbivores to Arctic ecosystems. Dr. Church removes a small vial containing mammoth DNA sequences that have been reconstructed from fragments recovered from permafrost specimens across Siberia. The genetic information in this vial represents the culmination of decades of research into mammoth biology and evolution, providing the blueprint necessary to engineer cells that carry mammoth genes instead of elephant genes. This is not science fiction, but applied genetic engineering using techniques that have become routine in medical research and agricultural biotechnology. The Mammoth de-extinction project began with the completion of the mammoth genome sequence in 2015, which provided researchers with a comprehensive catalog of genetic differences between mammoths and their closest living relatives, Asian elephants. Comparison of these two genomes revealed approximately 1.4 million genetic variants that distinguish mammoths from elephants, including crucial differences in genes controlling cold adaptation, fat metabolism, and hair growth that enabled mammoths to survive in Arctic environments. The technical approach to mammoth resurrection involves using CRISPR gene editing technology to systematically modify Asian elephant cells by replacing elephant gene variants with mammoth versions recovered from ancient DNA sequences. This process creates hybrid cells that carry mammoth genes in elephant cell nuclei, producing organisms that would have mammoth characteristics while retaining the basic elephant developmental program needed for successful reproduction. The first, Dr. Iriana Haisali, demonstrates the gene editing process using cell cultures derived from Asian elephant tissue samples. Individual elephant cells are treated with CRISPR-guide RNAs and Cas9 proteins that cut elephant DNA at specific locations and replace elephant gene sequences with mammoth variants. The process must be repeated for dozens or hundreds of genetic changes to create cells that carry a sufficient number of mammoth genes to produce mammoth-like characteristics. The reproductive biology of elephants creates unique obstacles for mammoth de-extinction that do not exist for other de-extinction projects involving smaller animals with shorter generation times. Elephant pregnancies last 22 months, sexual maturity is not reached until 15 years of age, and females produce offspring only every 4 to 6 years throughout their reproductive careers. These biological constraints mean that testing mammoth genetic modifications through traditional breeding approaches would require decades or centuries to evaluate success. Dr. Ben Lamb, co-founder of Colossal Biosciences, explains the company's timeline for producing the first living mammoth-elephant hybrid, acknowledging the enormous technical challenges while expressing optimism about eventual success. The company's current projections suggest that the first hybrid embryos could be produced within the next 5 years, with successful pregnancies possible within the following decade if artificial reproductive technologies continue to advance at current rates. However, the technical challenges of producing living mammoth hybrids represent only the beginning of the larger challenge of mammoth de-extinction. Even if genetic engineering succeeds in creating viable animals with mammoth characteristics, numerous additional obstacles must be overcome before mammoth de-extinction can be considered successful from conservation and ecological perspectives. The behavioral challenges facing resurrected mammoth populations may prove more difficult to solve than the genetic engineering required to create them. Mammoths were highly social animals that learned crucial survival skills through cultural transmission from experienced matriarchs who remembered traditional migration routes, feeding areas, and seasonal behavior patterns. Mammoth-elephant hybrids born in laboratories will lack this cultural knowledge and may be unable to survive in wild environments without extensive human intervention. The habitat requirements for mammoth populations create additional challenges that extend far beyond genetic engineering and reproductive biology. Historical mammoth habitat has been transformed by 10,000 years of environmental change, human development, and ecosystem succession that has replaced mammoth steppe with forests, agriculture, and human settlements. Successful mammoth reintroduction would require massive habitat restoration efforts that could cost billions of dollars and require international cooperation across multiple countries. The Pleistocene Park experiment conducted by Sergei Zimov in northeastern Siberia provides a potential model for mammoth habitat restoration while demonstrating both the possibilities and limitations of ecosystem reconstruction efforts. Zimov's research has shown that reintroducing large herbivores, including horses, bison, and reindeer to Arctic grasslands can partially restore mammoth steppe vegetation by preventing forest encroachment and maintaining grassland productivity through grazing pressure. However, the scale of habitat restoration required for viable mammoth populations far exceeds current conservation efforts and would require coordinated international action to establish protected areas encompassing millions of square kilometers across multiple countries. The political and economic challenges of such large-scale habitat restoration may prove more difficult to overcome than the scientific challenges of mammoth genetic engineering. The ethical debate surrounding mammoth de-extinction has intensified as the project has moved from theoretical possibility to practical reality, with critics arguing that resources devoted to mammoth resurrection would be better spent protecting currently endangered species that still have viable wild populations. Conservation biologists point out that the cost of mammoth de-extinction could fund protection efforts for hundreds of threatened species that could be saved through conventional conservation approaches. Animal welfare concerns represent another dimension of the mammoth de-extinction debate, with critics arguing that mammoth genetic engineering experiments will inevitably involve animal suffering as researchers attempt to perfect reproductive technologies and test genetic modifications. Failed embryo transfers, miscarriages, and developmentally abnormal offspring are likely outcomes during the development phase of mammoth de-extinction, raising questions about whether the potential benefits justify experimental costs. The genetic integrity of resurrected mammoth populations presents additional ethical and practical challenges that distinguish de-extinction projects from traditional conservation efforts. Mammoth-elephant hybrids will be genetically novel organisms that have never existed in nature, carrying combinations of mammoth and elephant genes that may produce unpredictable biological characteristics. These genetic chimeras will be neither true mammoths nor true elephants, but artificial creatures designed by human genetic engineering. Dr. Beth Shapiro argues that de-extinction should be evaluated not as a perfect resurrection of extinct species, but as a form of ecological restoration that could provide environmental benefits regardless of genetic authenticity. From this perspective, mammoth-elephant hybrids that can perform ecological functions similar to historical mammoth populations could help restore Arctic ecosystems even if they are not genetically identical to their extinct predecessors. The climate change mitigation potential of mammoth de-extinction represents one of the most compelling arguments for pursuing the project, despite its technical challenges and ethical concerns. Research suggests that large herbivore populations in Arctic regions could help preserve permafrost and reduce greenhouse gas emissions by converting forests and shrublands back to grasslands that reflect more solar radiation and maintain colder soil temperatures. Mathematical models of mammoth ecosystem impacts suggest that successful reintroduction of large herbivore populations to Siberian grasslands could delay permafrost melting by decades or centuries, potentially preventing the release of massive quantities of carbon dioxide and methane that are currently locked in frozen soils. If these projections are accurate, mammoth de-extinction could provide climate benefits that justify its costs as a geoengineering project rather than a conservation effort. However, critics argue that relying on de-extinction for climate change mitigation represents a dangerous distraction from more immediate and reliable approaches to reducing greenhouse gas emissions. The uncertainty surrounding mammoth de-extinction success rates and timelines makes it an unreliable foundation for climate policy, while proven mitigation strategies, including renewable energy development and fossil fuel reduction, remain underutilized. The international legal framework for de-extinction remains undeveloped, creating potential conflicts over ownership, responsibility, and regulation of resurrected species that cross national boundaries. Mammoth populations would naturally migrate across multiple countries in the Arctic, raising questions about which governments would have jurisdiction over de-extinction populations and responsibility for any ecological or economic damages they might cause. The intellectual property implications of mammoth de-extinction create additional legal complexities that could affect the long-term success of conservation efforts. Companies investing in de-extinction technology may seek patent protection for genetic modifications and reproductive techniques, potentially creating proprietary ownership of resurrected species that conflicts with traditional concepts of wildlife conservation and public ownership of natural resources. The timeline for mammoth de-extinction success remains highly uncertain despite optimistic projections from researchers and companies involved in the project. Conservative estimates suggest that producing viable mammoth-elephant hybrids will require at least 2 decades of additional research and development, while establishing self-sustaining wild populations could take many additional decades if it proves possible at all. Alternative approaches to mammoth de-extinction continue to be explored by research teams working on different aspects of the technical challenges. Some researchers focus on developing improved DNA recovery and sequence reconstruction techniques that could provide more complete mammoth genomes for gene editing. Others work on artificial gamete production that could eliminate the need for elephant surrogate mothers by creating mammoth sperm and eggs entirely through laboratory techniques. The partial de-extinction approach represents a potentially more achievable alternative to complete mammoth resurrection, focusing on creating elephant populations with selected mammoth characteristics rather than attempting to produce genetically complete mammoth replicas. This approach could achieve ecological benefits of mammoth reintroduction while avoiding some of the technical challenges associated with comprehensive genome editing. Dr. Vincent Lynch explores the evolutionary implications of mammoth de-extinction, pointing out that resurrected populations will inevitably evolve away from their engineered starting points as they adapt to contemporary environmental conditions that differ from Pleistocene mammoth habitats. De-extinction should therefore be viewed as creating evolutionary starting points rather than permanent resurrections of historic species. The long-term success of mammoth de-extinction will ultimately depend on factors that extend far beyond genetic engineering and reproductive technology. Climate change, habitat availability, human population growth, and international cooperation will all influence whether resurrected mammoth populations can establish sustainable wild populations that provide lasting ecological benefits. The question of whether justice can be restored for mammoth extinction may ultimately depend not on the technical success of de-extinction efforts, but on humanity's broader commitment to preventing future extinctions and protecting the species that still survive in an increasingly human-dominated world. The Mammoth case was solved through forensic investigation that revealed the complex causes of prehistoric extinction. The lesson from that investigation is that prevention remains more effective than resurrection, making the protection of currently endangered species the most important form of justice that can be provided for the victims of the ongoing sixth mass extinction. The cold case files stored in the basement archives of the Global Extinction Forensics Laboratory contain thousands of unsolved investigations that span the last 50,000 years of human expansion across the planet. Each file represents a species that vanished under circumstances that bear disturbing similarities to the mammoth extinction case, creating a pattern of evidence that suggests the same perpetrators have been operating continuously since the late Pleistocene. Dr. Anthony Barnosky opens another file as he begins the systematic review of extinction cases that could provide crucial intelligence for preventing future crimes against biodiversity. The Australian megafauna extinction case file contains evidence from a crime scene that predates mammoth extinction by 30,000 years, but follows an almost identical pattern of evidence. 45,000 years ago, Australia supported a diverse ecosystem of giant marsupials, including diprotodonts, the size of rhinoceros, kangaroos standing 3 meters tall, and marsupial lions that were among the most powerful predators ever to evolve. Within 5,000 years of human arrival, 90% of large vertebrate species had vanished from the continent, leaving behind only their smaller relatives and a simplified ecosystem that persists today. The forensic analysis of Australian megafauna extinctions reveals the same combination of climate change and human hunting pressure that eliminated mammoth populations, but with different timing and environmental conditions that provide additional insights into the extinction process. Human populations reached Australia during a period of relative climate stability, eliminating the possibility that environmental change alone could explain the rapid collapse of megafauna populations that had survived previous climate cycles. The archaeological evidence from Australian extinction sites shows clear signatures of human predation on megafauna species, including kill sites with stone tools, fire-altered landscapes that would have concentrated prey animals in smaller areas, and rock art depicting hunting scenes with giant marsupials. However, the speed and completeness of Australian extinctions suggests that factors beyond direct hunting were involved in eliminating species that were distributed across an entire continent. Dr. Gifford Miller presents evidence from amino acid racemization dating of emu eggshells that documents the precise timing of ecosystem collapse across different regions of Australia. The data shows that megafauna populations crashed within centuries of human arrival in each region, with extinctions spreading across the continent at rates that match human population expansion rather than environmental changes or disease outbreaks. The Madagascar extinction case provides another example of human-mediated extinction that occurred much more recently and under environmental conditions that were completely different from those faced by mammoths or Australian megafauna. When humans arrived on Madagascar approximately 2,000 years ago, the island supported a unique fauna of giant lemurs, elephant birds, and hippos that had evolved in isolation for millions of years. The Madagascar extinctions occurred during a period of stable climate conditions and involved species that had no previous exposure to human hunting pressure or introduced diseases. Despite these differences from earlier extinction events, the pattern of species loss followed the same rapid timeline that characterized mammoth and Australian megafauna extinctions, with most large vertebrate species disappearing within a few centuries of human contact. The forensic investigation of Madagascar extinctions reveals evidence for multiple human impacts that work synergistically to eliminate native species. Archaeological sites contain butchered bones from elephant birds and giant lemurs, while pollen records document widespread forest clearing that eliminated critical habitat for arboreal species. Introduced diseases and competition from domestic animals provided additional mortality pressures that accelerated extinction processes. The Pacific Island extinction cases represent perhaps the most extensive crime scene in the extinction investigation files, with evidence for human-caused extinctions on hundreds of islands across the Pacific Ocean, spanning thousands of years of Polynesian expansion. Each island colonization event was followed by the rapid elimination of flightless birds, large reptiles, and endemic mammals that had no evolutionary experience with human predation. The Hawaiian extinction case illustrates the pattern of island extinctions with particular clarity because of excellent archaeological preservation and detailed ecological records. Before Polynesian arrival, approximately 1,500 years ago, Hawaii supported numerous endemic bird species, including flightless geese, ibises, and rails that had evolved in the absence of mammalian predators. Archaeological excavations reveal that most of these species disappeared within centuries of human settlement. The forensic analysis of Hawaiian extinctions reveals evidence for multiple extinction mechanisms operating simultaneously. Direct hunting eliminated large birds that provided significant food resources for human colonists. Habitat destruction through forest clearing and fire removed nesting sites and food sources for smaller species. Introduced rats, pigs, and dogs created predation pressure on ground-nesting birds and reptiles that had no defenses against mammalian predators. The Caribbean extinction cases provide additional evidence for the destructive impact of human colonization on island ecosystems, with numerous islands losing endemic species following human arrival. The timing of these extinctions spans thousands of years from early Amerindian colonization through European contact, demonstrating that extinction vulnerability is not limited to particular human cultures or technologies. The New Zealand extinction case represents one of the most recent and well-documented examples of human-mediated extinction, occurring entirely within the last 1,000 years and providing detailed insights into extinction processes. When Polynesian settlers reached New Zealand approximately 700 years ago, they encountered a unique fauna of flightless birds, including 9 species of moa, ranging from turkey-sized to ostrich-sized animals. The MOA extinction investigation reveals clear evidence for rapid human impact on New Zealand ecosystems, with most moa species disappearing within 3 centuries of human arrival. Archaeological sites contain abundant moa bones showing butchering marks, while early European accounts describe landscapes littered with moa bone beds, indicating massive hunting pressure during the initial colonization period. However, the New Zealand case also demonstrates that human hunting alone was insufficient to explain the completeness of moa extinctions. Habitat destruction through forest burning eliminated critical habitat for moa populations, while introduced Pacific rats attacked moa eggs and chicks in ground nests. The combination of adult mortality from hunting and reproductive failure from predation and habitat loss created demographic conditions that made moa extinction inevitable. The North American megafauna extinction case provides the closest parallel to mammoth extinction in terms of timing, environmental conditions, and species affected. 13,000 years ago, North America supported a diverse megafauna, including giant ground sloths, saber-toothed cats, American lions, short-faced bears, and horses that disappeared at approximately the same time as mammoth populations. The forensic investigation of North American extinctions reveals the same pattern of evidence that characterized mammoth extinction, with rapid species loss occurring during a period of climate change and initial human colonization by Clovis populations. The synchrony of extinctions across different species and regions suggests that common factors were responsible for ecosystem-wide collapse rather than species-specific vulnerabilities. The Clovis archaeological record provides direct evidence for human predation on mammoth and other megafauna species, but the extent of hunting pressure remains controversial among researchers. Mathematical models suggest that sustainable hunting by small human populations could have eliminated megafauna within centuries, while critics argue that archaeological evidence is insufficient to support such rapid and extensive hunting impacts. The South American megafauna extinction case occurred slightly later than North American extinctions, but involved many of the same genera and followed similar patterns of rapid species loss during periods of climate change and human expansion. Giant ground sloths, glyptodonts, and native horses disappeared from South America during the late Pleistocene and early Holocene, leaving behind only smaller relatives that survive today. The current African elephant extinction case represents the closest modern parallel to historical mammoth extinction, with populations declining from millions of individuals a century ago to fewer than 400,000 today. The forensic investigation of elephant population decline reveals the same combination of habitat loss and hunting pressure that eliminated mammoth populations, but occurring over decades rather than millennia. The ivory trade provides direct evidence for commercial hunting pressure that targets adult elephants for their tusks, creating demographic impacts that are similar to those that may have affected mammoth populations. Poaching pressure is particularly severe for female elephants, which carry smaller tusks that are easier to transport but represent the reproductive core of elephant populations. Habitat fragmentation represents another factor affecting modern elephant populations that parallels the environmental changes that stressed mammoth populations during the late Pleistocene. African elephant populations are increasingly restricted to protected areas surrounded by human development, creating isolated populations that are vulnerable to genetic problems and environmental catastrophes. The rhinoceros extinction crisis provides additional insights into the vulnerability of large herbivores to combine human and environmental pressures. 5 rhinoceros species remain from a historically diverse group that once included dozens of species across Africa and Asia. Most surviving populations number in the hundreds or low thousands of individuals, making them vulnerable to extinction from multiple causes. The forensic analysis of rhinoceros population decline reveals the same pattern of synergistic factors that eliminated mammoth populations. Habitat loss reduces carrying capacity and fragments populations into isolated groups. Poaching for horn trade creates direct mortality pressure that particularly affects adult breeding animals. Climate change alters habitat quality and water availability in ways that stress populations already facing other mortality factors. The polar bear extinction investigation provides a modern example of climate change-mediated extinction that closely parallels the environmental challenges faced by mammoth populations at the end of the Ice Age. Arctic sea ice that polar bears depend on for hunting seals is disappearing at unprecedented rates, forcing populations to spend longer periods on land where food is scarce. The forensic analysis of polar bear population trends reveals declining body condition, reduced reproductive success, and increased mortality that are directly linked to habitat loss from climate change. Some populations have already declined significantly, while others remain stable but face inevitable habitat loss if current warming trends continue. The coral reef extinction crisis represents an example of ecosystem-wide collapse that provides insights into how environmental change can eliminate entire biological communities. Rising ocean temperatures, acidification, and pollution have caused widespread coral bleaching and death that has eliminated reef ecosystems across large areas of the tropics. The pattern of coral reef collapse shows how environmental stress can trigger cascading extinctions that affect entire food webs rather than individual species. Fish populations that depend on coral reefs for shelter and food sources decline following coral death, while predators that feed on reef fish experience secondary population impacts that can lead to regional extinctions. The amphibian extinction crisis provides evidence for disease-mediated extinctions that support the disease hypothesis for mammoth extinction. Chytrid fungus has spread across multiple continents within the last few decades, causing population collapse and extinction in hundreds of frog and salamander species that had no evolutionary experience with this pathogen. The forensic investigation of chytrid-mediated extinctions reveals rapid population collapse that occurs within years of pathogen arrival, demonstrating that disease can cause extinction rates that are far faster than those associated with habitat loss or direct mortality from human activities. The global spread of chytrid fungus shows how modern transportation can facilitate pathogen dispersal across biogeographic barriers that historically prevented disease transmission. The bat extinction crisis caused by white-nose syndrome provides another example of disease-mediated population collapse that demonstrates the potential for introduced pathogens to cause rapid species decline. This fungal disease has killed millions of bats across North America within 2 decades of its apparent introduction from Europe, causing population declines of more than 90% in some species. The pattern of white-nose syndrome spread shows how disease can affect multiple species simultaneously, while showing varying impacts based on species-specific susceptibility and behavioral factors. Some bat species appear to be developing resistance to the pathogen, while others face continued population decline that may lead to extinction if effective treatments are not developed. The monarch butterfly extinction investigation reveals how multiple factors can combine to threaten species that were historically abundant and widespread. Monarch populations have declined by more than 80% during the last 2 decades due to habitat loss from agricultural expansion, pesticide use that kills caterpillars and reduces milkweed host plants, and climate change that affects migration timing and overwintering survival. The monarch case demonstrates how widespread species can become extinction vulnerable when multiple threats combine to exceed population resilience. Individual factors might be manageable separately, but their synergistic effects can drive even abundant species toward extinction within decades rather than centuries or millennia. The lessons learned from these modern and historical extinction cases provide crucial intelligence for developing prevention strategies that could protect currently threatened species from following mammoth populations into extinction. The forensic investigation of multiple extinction events reveals consistent patterns that can guide conservation efforts and help identify species that are most vulnerable to extinction under current conditions. The most important lesson from the extinction investigation files is that prevention is far more effective and less expensive than restoration efforts, whether through de-extinction technology or ecosystem reconstruction. Once species are eliminated from ecosystems, the complex ecological relationships that supported them are disrupted in ways that can take centuries or millennia to restore, even if the original species can be returned. The investigation files also reveal that extinction vulnerability is not randomly distributed among species, but follows predictable patterns based on life history characteristics, ecological requirements, and human impact susceptibility. Large-bodied species with slow reproduction rates are consistently more vulnerable to extinction than smaller species with rapid population growth. Species with specialized habitat requirements are more vulnerable than generalists. The timing patterns revealed by extinction investigations show that species loss often occurs rapidly following initial human contact or environmental change, suggesting that early intervention is crucial for preventing extinctions. Once population decline begins, multiple feedback effects can accelerate extinction processes in ways that make recovery increasingly difficult, even if initial stressors are removed. The geographic patterns of extinction reveal that isolation increases extinction vulnerability, whether through natural geographic barriers or human-created habitat fragmentation. Island species are consistently more vulnerable to extinction than mainland species, while small isolated populations face higher extinction risks than large connected populations. The modern extinction cases demonstrate that current species loss rates are far higher than historical background extinction rates and are approaching levels that characterized major mass extinction events in the geological past. The current situation represents an ongoing extinction crisis that requires immediate intervention to prevent the loss of thousands of additional species within the coming decades. The prevention strategies developed from extinction investigation findings focus on addressing the root causes that have driven species to extinction throughout human history. Habitat protection and restoration efforts target the environmental requirements that species need for long-term survival. Anti-poaching enforcement addresses direct human predation pressure that can rapidly eliminate vulnerable populations. Climate change mitigation represents a crucial component of extinction prevention that addresses one of the primary factors identified in both historical and contemporary extinction cases. Reducing greenhouse gas emissions and developing adaptation strategies could prevent climate change-mediated extinctions that would otherwise eliminate thousands of species during the coming century. Disease monitoring and quarantine protocols represent another prevention strategy that addresses the pathogen introduction risks that may have contributed to historical extinctions and continue to threaten modern species. Early detection systems and rapid response protocols could prevent disease outbreaks from causing the kind of rapid population collapse seen in chytrid-affected amphibians and white nose syndrome-affected bats. Genetic management represents a prevention strategy that addresses the inbreeding and genetic drift problems that affected small mammoth populations and continue to threaten modern endangered species. Maintaining genetic diversity through population management and genetic rescue efforts could prevent the genetic meltdown that ultimately eliminated the last mammoth populations on isolated islands. The international cooperation required for effective extinction prevention represents one of the greatest challenges facing modern conservation efforts. Many threatened species cross national boundaries during migration or have populations distributed across multiple countries, requiring coordinated conservation efforts that can be difficult to achieve given political and economic differences between nations. The economic incentives that drive many extinction-causing activities represent another major challenge for prevention efforts. Poaching, habitat destruction, and pollution often occur because they provide short-term economic benefits that outweigh conservation values and market-based decision-making. Creating economic incentives for conservation requires fundamental changes in how natural resources are valued and managed. The public awareness and political support needed for large-scale conservation efforts remain insufficient, despite growing recognition of extinction risks and biodiversity loss. Developing effective communication strategies that convey the urgency of extinction prevention while providing realistic solutions represents a crucial component of conservation efforts. As the extinction investigation files are closed and returned to their archive storage, the evidence they contain provides both warning and hope for the future of biodiversity conservation. The warning is clear. Human activities continue to drive species toward extinction at unprecedented rates through the same mechanisms that eliminated mammoth populations 10,000 years ago. The hope lies in humanity's growing understanding of extinction processes and development of intervention strategies that could prevent future losses if implemented quickly and effectively. The mammoth extinction case was solved through careful forensic investigation that revealed the complex causes and consequences of prehistoric species loss. The lesson from that investigation extends far beyond understanding what happened to mammoths to provide guidance for protecting the species that still survive in an increasingly human-dominated world. The case files remain open on current extinction crises, waiting for the intervention that could write different endings to stories that otherwise will conclude with the same tragic verdict that closed the mammoth case 10,000 years ago.